So good afternoon, friends. Honorable Founder President, Dr. Ashok Chauhan ji, our very dear Chancellor, Dr. Atul Chauhan ji, and all the members of Amity Fraternity, and those invitees, guests who have joined here in our this beautiful Right to Mood Court, and also online. So I welcome each and every one of you. Dr. Newton, please join your seat. Particularly, I extend a warm welcome to our Honorable Founder President, who has joined us right from the beginning and who has also given a lot of guidance in organizing this concept of path-breaking research. And the whole initiative came from Dr. Ashok Chauhan, the Founder President, who always makes new paradigms, new initiatives, research clusters, path-breaking research. These are all very, very novel approach to give an impetus to research innovation in our campuses. And for him, it is the research and innovation is the first priority. He lives innovation, needs innovation. So, Founder President, we are so grateful. Whatever Amity has reached today in terms of the positions at the national, global level, not only in academics, but also in research, his contribution is seminal, pivotal, and thank you so much for giving that kind of leadership here for research innovation. I also welcome the experts who have joined us today in this path-breaking research. I will mention the experts' named speakers and also all the participants who are here online, offline. Research and innovation is the focus in Amity. Not only Amity universities, even in schools, we have given a focus. So right from the schools onwards, even preschools, we promote creativity, innovation, problem-solving abilities in our children. So the research and innovation is the fulcrum of our educational system in Amity Group itself. The Honorable Founder President, Madam Chairperson, the Chancellors, everyone gives a lot of importance and also impetus for research and innovation. That is how today Amity ranks very high in research and innovation. When I look at the research achievements in the last about a decade, because I have spent almost a decade here. Please mute your mic if you are online. Thank you so much. So when you look at, we have 12 universities in India, 18 campuses abroad, 28 schools. This is for the people who have come from outside, the experts who may know much, but then I wanted to supplement what they don't know. That today in all these institutions that we are giving a focus in research, 5 Ps, that is projects, patents, publications, PhDs, products and technology development. So this is the focus in all our research. So our research in Amity has to culminate in any one of them. It could lead to a product development, technology development, process development, or it could be solving a problem of a society, or it should be ultimately leading to a path-breaking research. That's what we are going to listen to today. This path-breaking concept, the Founder President deliberated with us, and then ultimately what we thought that we must project the brilliant science coming out of the portals of the Amity group of institutions, Amity universities. So we keep the track of all this in ASTIF, APSTIA, and also ADSI. These are the 3 pillars for research innovation. So we keep track of the talents emerging from various campuses in research and innovation. We have profiled these people. So we know their H index, I-10 index, how many projects they have done, how many publications they have made, and also what is the innovative research this individual has done. If the database is available with these 3 verticals. Then Founder President has also created a Thunder Force, S&T Thunder Force. This S&T Thunder Force is more than 2,000 people in all our campuses. So that gives the field effect when they start working in their own institutions. That field effect also percolates down to all campuses. So we have those S&T Thunder Force, which is more than 2,000 in our campuses, who give all this research, innovation focus. Then we have also established Amity Global Research Hub. We have in our Long Islands in New York, we have a big campus founder president has acquired, and Chancellor Dr. Asim Chauhan is driving that mission over there. Amity Global Research Hub. That means we would like to connect all the Amity group of institutions in India with selected institutions across the globe, particularly in US. That is why we have established it in the Long Islands of New York. So this global research hub, again, will create new paradigm in terms of bringing the institutional connect, industry connect, and ultimately a force multiplier in the outcome of our research. So the philosophy of research in Amity Group, as I said, the first, it has to be leading to a deliverable. Second is the deliverable has to be sustainable. So we look into sustainability of these research and innovation and its outcomes. And the next parameter we look at for our research outcome is the affordability. Whatever we do, it should benefit a larger cross section of the society. So if you develop a product or a technology, it should be affordable, accessible to the people. So we keep that as another focus when you look at the Amity research and innovation. The next part is it should lead to the path-breaking research. It's something unique. It is not the run-of-the-mill to confirm somebody has done in a different country and we want to confirm, reconfirm, or negate what their findings are. But then we want to do always something, a niche area, disruptive technologies, path-breaking research outcome is always aimed at by our people. So this is yet another session. We have already completed 3 series of this session, path-breaking research presentations by our ASTIF has organized in the last year. Now, this is the first one in this year, that is 2023. This is the first one, and where we have picked up 4 brilliant faculties, scientists who from Amity Group, that they have done remarkably well. 3 are from this campus, Amity University, Uttar Pradesh, Noida, and we have 1 from Amity University, Haryana, which is also doing a wonderful job. So we have 4 of these very distinguished people, including Dr. Mona Lisa Mukherjee, who is the director of Amity Institute of Click Chemistry and Research Studies, Dr. Durgesh K. Tripathi, Amity Institute of Organic Agriculture, then Dr. Malika Chatterjee from Amity Institute of Neuropsychology and Neuroscience, and we have Dr. Ravi Datta Sharma from Amity University, Haryana. He is professor at the Amity Institute of Integrative Science and Health from Amity University, Haryana. So these are the 4 presentations which are going to be made. And we have also picked up the experts. After the presentation, each presentation, there will be 2 experts from outside Amity who would look at this. And so also they will motivate, inspire our presenters, our faculties, our scientists, but also give critical assessment of the quality and also how do we take it forward, ultimately to handhold the investigator or the inventor, and how do we handhold this pathfinding, the breaking finding to a conclusion, to a culminating in the deliverable, perhaps a deliverable. So this is what is the function of the experts who are here. Thank you so much for all the experts who have joined offline, online, over here, because you have a big role, because we value your advice, guidance, and also this going to connect with them in a sustainable manner. Not only this, looking at this presentation here, then we would request the faculties to connect with you and also handhold, take the guidance from you periodically so that this research, path-breaking research, leads to some impactful outcome on the society itself. So the experts are from various institutions, starting from Vadera. Vadera is a very outstanding scientist from DRDO. He was in DRDO, and he was the director of a very strategic laboratory, Defense Lab Jodhpur, which is on the western side. But now he is the deputy director at the Amity Institute of, Indian Institute of Technology, Jodhpur. So I visited that IIT with a big delegation, including Dr. Gopal Bhushan, who accompanied me. And then finally, we thought the outstanding work after he moved out from Defense Lab Jodhpur. He's doing a great job in the physics department. He is the professor and driving many missions with the experience in DRDO. He's enriching the research over there. So Dr. Vadera, welcome to you, and we are all looking forward to listen to you. We will also have listened to the next expert, Dr. Himadri Bohidar, and he is professor in the School of Physical Sciences at JNU. Thank you, Professor, for joining. And then we have Dr. Rupesh Deshmukh, Associate Professor from in the field of biotechnology from Central University of Haryana. We have Dr. Shivesh Sharma, MNNIT, National Institute of Technology, Allahabad, and Dr. Usha Panjwani, who is in the DIPAS, Defense Institute of Physiology, heading the neurophysiology, just superannuated now. Professor Pankaj Seth from NBRC, whom we can see it on the screen. And I'm so grateful to all of you. And also we have the other experts who have joined us. Dr. Vijay Mishra. Good afternoon, Dr. Andrew. And I'm so happy, grateful that you could join us. Dr. Vigyan Mishra, Chief of Lab from Newburgh, who is here, physically present here, Newburgh Diagnostic. Professor Andrew Lin, he is from Jawaharlal Nehru University, School of Computational and Integrative Science. Thank you very much for joining. Good afternoon. I'm so grateful that you could join. Dr. Vishal Nanavati from the DBT Ramalingasamy Fellow and Consultant and Senior Scientist for Genomic Medicine. So I think we have wonderful experts now here who are going to also critically give some suggestions, advice, guidance to take forward these wonderful research. And the topic, if you see, these are all very important topics in which the 4 path-breaking research are going to be presented. The first one is going to be self-assembly and nanostructure. So that is the area, the domain area in which the presentation would be made. Then we have neurobehavior models. When you study the pathophysiology of the brain and its functions, brain in health and disease, what are the models we can use for the neurobehavior, which will be affected in the pathological conditions? So that is the next area. The third area is transcriptional the biomarkers. Biomarkers is assuming a great relevance today, in particular in diagnostics of many diseases. So, and also prognosis. So we have one of the presentation would be related to this domain. Lastly, we have decoding of decoding silicon potentials in plants. So this is going to be the 4th presentation. This is from the organic agriculture side. So you can really be here for intellectually enriching experience from these 4 presentations and also comments from the experts who have joined us here. And let us start with the first one, Dr. Mona Lisa Mukherjee. I have pleasure in introducing Dr. Mona Lisa. She is the director of the Amity Institute of Click Chemistry and Research Studies, professor in Amity Institute of Biotechnology in the Amity University, Uttar Pradesh, Noida, and founder president when Dr. John Moses visited from US, initially from Nottingham, and he spoke on click chemistry. So immediately they said we should now launch a major initiative, and Dr. Mona Lisa was appointed as the director. She has done great wonders, and she's a IIT Delhi product. She's PhD from IIT Delhi in 2006, and she is a National Scholarship Awardee, Young Scientist Awardee, and also UK India Distinguished Visiting Scientist Award in 2011. She is Fellow of the Royal Society of Chemistry, London in 2021, just a couple of years ago. Then her focus is in the field of biomaterials or materiobiology. Is the next founder president. Welcome, founder president, sir. We could see you on the screen. Let's clap for founder president. Thank you for joining. I know how busy he is, hopping from one meeting to the other online. Thank you, founder president. It's a great inspiration to see you. Thank you so much. And then Mona Lisa also has published a large number of papers, and she has guided 7 PhD students. Her publications are in Journal of American Chemical Society, ACS Nano, Nano Today, Material Horizon, ACS Applied. She has submitted proposals. She's an honorary professor in Telangana University and many, many more accomplishments. Let us greet. And before she starts it, I would request Founder President to say a few words and give your blessings, and then we will start the proceedings. Over to you, Founder President. Okay, Moderator, you can start.
So I can start. So good afternoon, everyone. It's a really nice opportunity for me. First of all, I try to do good research. This is my passion. But as of now, I don't know it is a path breaking or not until unless it's come to the market. Of course, Meenakshi is trying her best.
I think Founder President Mona, just can you wait for a few minutes? Founder, would you like to make a few remarks now? Okay. Right. Go ahead, Mona.
Okay. So it's path breaking or not, I don't know. But yeah, we are trying our level best to make it, to commercialize the things, to bring it to the society. And of course, Meenakshi is trying her best to get it into the market with the help of High Media or Thermo. And hopefully we have done a couple of meetings based on that. Hopefully it will soon come to the market. So we are hoping for that. So an unexplored avenue of self-assembly in the genesis of exotic nanostructures. I am a chemist by my master's specialization. By my PhD, I am a biomedical engineer. So I have done some of the mimicking biomimetics, catalysis, and engineering during my PhD tenure in IIT Delhi. So I have done my work in mimicking cytochrome P450 and nitric oxide synthesis. And by doing that work, I have delineated the pathway, how this xenobiotics do their work, and came up with a mechanism which was most cited, and it was published in Journal of American Chemical Society within 2 years of my PhD tenure in IIT Delhi. So today I will speak about what work I have done in Amity. So last 13 years I am here in Amity. I started my work with Amity Institute of Biotechnology. So the genesis is with Amity Institute of Biotechnology. And then I moved to Nottingham University as a distinguished fellow to work with John Moses. And there I got connected to Barry Sharpless. Barry Sharpless, who is the Nobel laureate in chemistry, in the history of chemistry in the entire world, the person who got the Nobel Prize for twice for Sharpless epoxidation reaction, and the last one, which was given to him, bestowed on him last year for click chemistry. So at that time, it was very starting phase for me to learn about click chemistry. So John speaks, he learned more about nano from me, rather what I learned more click chemistry from him. So that bonding started. He visited us. He was bestowed as chair professor from Amity Institute of Biotechnology, an honorary professor from Amity Institute of Nanotechnology. The work went on. We tried to submit some of the projects. I have completed 6 projects. Among them, although Indo-UK project, he was one of the part. And we have started working in that area, learning of biomimetic catalysis, and then moved ahead for working in the area application part, more related to colon cancer drug delivery for stem cells, which I will be showing in my slides. So what I like to say before I start my presentation is self-assembly. So before I go to the presentation in details, I will tell you a snapshot what the presentation is all about, because it is not possible to speak about all my work in this 25 minutes or 20 minutes of time which I have been given today. Otherwise, it would have been a plenary speaker. So self-assembly, you all know, self-assembly is a process to make organized structure from atoms, molecules, or part of the molecules without human interventions. So without human interventions, when we tell, that means these are specific or local interactions. So there may be van der Waals interactions, electrostatic, there may be electromagnetic interactions. So without any human intervention. So self-assembly is very natural. In living organism and all this, we see the self-assembly very common. So I will be talking about graphene quantum dot because I'm going to tell you about exotic nanostructures. What do you mean by exotic nanostructures? Unnatural, unseen, some new, exciting nanostructures. So graphene quantum dots, how that is going to give rise to exotic nanostructures with the help of polymers. So that few minutes I will be taking you to the path of that area where new exciting exotic nanostructures will be shown in my slides. So graphene quantum dots are harbingers of paradigm shift. You all know graphene, that is a 2D system. Even 3D, we can go for 3D. But why I'm coming to graphene quantum dot? Because of the quantum confinement, restricted movement of the electrons confined within the dot in the discrete energy levels. And when it absorbs light, then depending on the size, shape, they can give rise to different wavelengths. So they can be used as a fluorescent molecule. Is it restricted to that only? No. It has some very good optical and electronic properties and also directional properties. So graphene quantum dots are harbingers of paradigm shift, which revitalizes the colloidal puzzle by giving shape and size to the material design palette. So self-assembly is ubiquitous in nature. It is very common. You have seen that small molecules self-assemble to give a big molecule that is an organized way, right? So it is very common or ubiquitous in nature. But how or the extent this molecular Legos can be engineered reminds us that we are still apparentizing polymeric carpenters. In this quest to unlock the exotic nanostructures, we came up with the strategy to use graphene quantum dots for polymerizations, using them as a filler, nanofillers for free radical mediated aqueous copolymerizations. Now, extensive polymer grafting takes place in the confined space because it is a graphene quantum dot, 0 dimension. So extensive polymer grafting takes place and which bolsters thereby the crystalline phase. And because of this crystalline phase, what happens? There is a steric interaction between the loops of the polymer and graphene quantum dot. And as a result, it gives rise to an interlaced equilateral triangle, which gives the shape of exotic nanostructure, kagome, the Japanese basket or basketry. But at higher concentration of graphene quantum dot, there is again an interesting thing. They form J aggregates, which I will be showing. And these J aggregates then helps the polymer to use or fine tune its property so that they can break the symmetry and they can form Janus exotic nanostructures. What is Janus? It is biphasic God. So all these morphological characteristics was envisaged with this research, and then with this type of structures, we went for applications, which was seen in rabbit model, and we have delivered 5-fluorouracil to the colon, and it was seen to give the best result among the literatures which are reported till date. So look deep into the nature, then you will understand better. Organisms are formed through self-assembly. Biological system elegantly modulate self-assembly with great precision. The origami of self-assembly being used in material science that emphasizes on initiating the generation of artificial platforms influenced by nature. So material science is basically a snapshot on the way from fundamental science to societal impact. So I'm working in the area of materiobiology. So what is self-assembly? One of the most wondrous aspects of life, which I told that all living organisms are formed through self-assembly without human intervention. They do because of local and specific interactions. Is it audible? There are four main components which allow self-assembly. Sound.
Mona, you can speak on this.
So I don't think the sound is there. So the structured particles, environment, driving force, binding force. This is just a small video. I want to show how self-assembly is taking place.
You can give the voice over.
Yeah, might be. Basically, how the atoms or the part of the molecules come together and they assemble to form an organized structure. So the hexagonal pattern, how it is done. As the water is stirred, the sticks begin to travel over us, flying down with the pair stuck together. The different particle shape self-assembled structure, again with the help of sticks. So that has been done. So already have discussed about this. Let me go to the next slide. Why exotic nanostructure formation so difficult? Because of the facile control over the spatial arrangements of nanoscopic building blocks in the bottom-up synthesis. What do you mean by bottom-up synthesis? Small molecules gathered together to make a big molecule. Suppose we are synthesizing carbon nanosheets and we are taking glycerol, we are taking sulfuric acid. These are the small molecules and we are putting it in the hydrothermal bomb where we get water as a media for reaction and the autogenous pressure of the hydrothermal bomb. So that autogenous pressure and temperature, if we go for low temperature hydrothermal carbonization or we go for high temperature hydrothermal carbonization, give rise to metastable state. Now what is metastable state? Suppose I'm dropping this. This is the excited state. I drop this. This is the ground state, right? We can get the properties of the ground state. We can get the properties of the excited state. What about the intermediate steps? So the intermediate steps are called metastable states. So this is the beauty which was invented by the chemist, and Professor C.N.R. Rao has done a lot of work in this area, and even Dr. Raju, that they have tried to bring out the metastable states which can inculcate new properties to the molecules for myriad applications. So that can give rise to even an exotic nanostructures. So high level of complexity exists with structural precision where independent control of polymer remains an unmet challenge, growing bottlenecks of precision assembly while designing well-ordered nanostructures across the lens. Then again, sub 10 nanometer range, low symmetry topologies, and controlled anisotropy. It's a very important word which I told in the start when I gave the snapshot of my presentation. Anisotropy, anisotropy. Anisotropy means directional property. Isotropy means it is homogeneous. So in all the direction, it has same properties. But anisotropy, based on a particular direction, you will have a particular property. So the anisotropy, orientation, topology, molecular functionality, hierarchical self-assembly are still at their infancy. So fabrication of exotic nanostructure using synthetic polymers for biomedical applications. For that, for every studies, you need to read a lot. If you do 5% work, you need to study 95%. So graphene carbon nanotubes in biomedical arena, friend or foe. For that, we went to study the identical routes to malignancy from asbestos and carbon nanotube with the current biology. Then we went to study with the nature nanotechnology promises, facts, challenges for carbon nanotubes in imaging and therapeutics. Then we went to study for the science paper, toxic potential of materials at the nano level, which is very important parameter because we want to go for the biomedical applications. Then we went for understanding the toxicity of carbon nanotubes because till date, people were speaking more about the carbon nanotubes. Then we went for studying functionalized single-walled carbon nanotubes as rationally designed vehicle for tumor-targeted drug delivery. And in doing that, we also came across this multi-walled carbon nanotubes functionalization with high molecular weight hyaluron significantly reduces the pulmonary injury. This is the most efficient polymer which is used because they mimic the ECM, extracellular matrix. That's why it was used in a bigger extent. Then, having all the studies done, I came up with a strategy, a nascent, a simple strategy where acrylic acid and DMA, PMA, these are both the monomers, they are used in 30% and 70% concentration. In a free radical mediated aqueous copolymerization, where timid is taken as an accelerator, APS, that is ammonium persulfate or sodium persulfate, is taken as an initiator to design this hydrogel nanotubes. Now, 30% of those who are chemists can really understand this, but still I'm trying to make everyone understand about 30% of DMA, EMA will react with 30% of acrylic acid to form a copolymer. So what about the remaining 40% of acrylic acid? The remaining acrylic acid will be converted into the polyacrylic acid. So this polyacrylic acid is highly soluble in water, or they are solubilizing, they are water loving, they form the corona. And the core is the hydrophobic, that is acrylic acid, which forms copolymer with the comonomer DMA-EMA, and that is the hydrophobic zone. So it will be a micellar structure. It is the hydrophobic and outside is the hydrophilic. And in this way, they try to make a sheet-like structure. Why? I told about mycelium, then where comes the sheet? Because of the structured water entrapped in the polymer matrix, they give rise to nanoconstriction. This nanoconstriction in the nanoresin helps the formation of the sheets. These sheets then buckle and they form a tubal architecture, and the water confined them form a helical pattern which has been seen with the help of TEM microscope. So this work was done by our lab in 2019. And this is the image. We have done this work. I want to tell you that we have done the reactions in every stage to identify at what stage these helical tubes are formed. The ice helix are formed. And we need to give the possible data, especially the morphology, the DFT studies, the MD simulations to support that, yes, it is the ice helix which is entrapped into the nanotubes. And this is the first report by our group. So we have done the reaction 4 hours, 8 hours, 10 hours, and 12 hours. We have taken the aliquots and we have done the TEM microscope where we see that stacked polymer nanosheets after 10 hours, indicating embryonic polymer nanosheets with lateral dimensions of nanometer range, picture-like architecture. If you see, this is a picture-like architecture. And a tubular structure is formed. Tubular structure is formed, rolling up of polymer nanosheets to form HNTs. This is the HNTs. And these are shown at low magnification. Then we went to see at higher magnification, higher, that is HRTM, and we saw very beautiful helix-like pattern. Now, are they helix? How they are formed? That has been discussed in our paper, and we have found that it is the water which is giving rise to the cyclooctane domains, and it is the 2D sheets which buckle out to form a cyclohexane moiety, which is seen from selected area diffraction pattern, giving rise it is not seen. Okay, giving rise. So this is the selected area diffraction pattern. So you can see that we are having a cyclooctane ring and outside the cyclohexane ring. So that cyclohexane ring is telling about the sheets and the water, how they are interacting with the NN-methyl group of DMA-EMA, and they are forming this helical pattern is seen here. And then again, we are confirming that the bundle of HNTs, the arrows indicates the helices entrapped with the HNTs, and the hexagonally structured eyes, which I said, the hexagonally structured eyes, TA micrograph, crumpled polymer nanosheets, disordered tubular architecture, and this. Now, why I said disordered? Because first of all, the question comes that we are doing at 40 degrees centigrade. Earlier, it was done, this kind of tubes, they generate at very, very high temperature. This is the first reaction where we have reported at 40 degrees centigrade. So why this is happening? Because of the nanoconstriction, the nano regime that is coming into the forefront and trying to make this kind of helical structures.
Mona Lisa, can you just move a little faster?
Okay.
We have other free presentations.
Okay. Then I will just give the snapshot of that. And so we have gone through the SEM, the beautiful polymer wrinkles, the clusters of HNTs. We are seeing braided structure, all taken from Amity University, from Dr. Jan's department, and I acknowledge his department for the same, for the beautiful SEM microscope, the unfurled knots, the hairpin turns, and the HNTs with the diameter of 62 nanometer and the hollow tubular architecture. So diabetic wound, perfect storm for a nick to become a life-threatening wound. People have done for 20 days, also done with 20 days using diabetic wound healing for using the bioglass and bioactive injectable hydrogels containing desferoxime and bioglass for diabetic wound healing. People have done with copper metal organic framework with folic acid, improved wound healing diabetes, but we have done within 16 days. So it is the structure, the exotic nanostructure, which is giving sustained release of water or sustained release of drug with the help of this helix and trying to keep the exudates away from the healing site and giving the diabetic healing. This has been done 3 eyes, and you will be very happy to know that this technology has been now taken by Jubilant Biosys, and they are trying to make the things in their laboratory, and they are trying to do the same thing if they can do for scarless healing of burn wounds. So this is the work which was published in Material Horizon, and I take the pleasure to tell that Aarti Singh, Rohan Bhattacharya, Adiba, Arun Kumar Sharma, Dr. Arun Kumar Sharma, who is sitting over here, has done the entire biological work. He was working with me as a postdoc and now a faculty and scientist doing great work. And this was also being shown in The Hindu by Prasad Rabindranath, and it was accepted as the best paper of Horizon Community Board by Material Horizon Royal Society of Chemistry. So let me move fast. Molecular self-assembly and drug delivery. So I'm not going to show the video. Polymeric patterning at nanoscale, which I said that graphene quantum dot, they try to give the shape and size to the colloidal puzzle and give rise to a particular exotic structures, which is Kagom and Janus, which I told you that I will be showing. So till date, why I'm showing these papers? Because till date, the work of Kagom generation was done with the help of metal. But first time we have done with graphene quantum dot without any metal templation to give rise to a Kagom structure. Because if metal is there, then toxicity will be still induced in the system. Now, going to Janus, that is a biphasic system of drug delivery. Janus was also known, but Janus, people used to prepare in oil emulsion, with high temperature, with organic molecules. So again, there is again a restriction to use those molecules for drug delivery. But here we have done with the help of graphene quantum dot as nanofiller, giving the Janus formation with this copolymer, with this polymer, and making them towards the Janus with the electrolytic complex differentiation, phase differentiation, because if we go for graphene quantum dot, they will try to react with acrylic acid as well as DMA, EMA. Now there will be electrolytic complexes formed in the media. These electrolytic complexes will come in closer proximity, and in presence of water, they will have a zone of separation. That zone of separation will give rise to the Janus biphasic system. So this work was done with graphene quantum dot.
Another 5 minutes?
Yeah, I'm trying, sir. Very sorry, I'm going very slow because I thought if you need to understand people, then you have to tell each ifs and buts of the research. So the challenges of graphene quantum dot, this also was done in our laboratory. We have done from molasses, so waste to best, with comprehensive utilization of the product. What does it mean? Leaving no human footprints. Even the byproduct was a sensor for sensing dopamine, but the product is highly fluorescent molecule which can go for imaging the HG2 cells. And these materials were used as filler for making this exotic and Janus structures. So I'm not going to the synthesis. This is the morphology. At lower concentration, it is homogeneously dispersed. But at higher concentration, they form J aggregates. Why this is so? Because the hydroxyl groups, the SO3H groups, and the carboxylate groups, they deprotonate. And as a result, they try to form an aggregates. And these J aggregates then play an immense role in forming the Janus nanostructure at higher concentrations. So again, this was the first report by us in green chemistry, where we said that the agro-industrial based molasses has cyclohexane domain as the major phase, but it gets converted to graphene hexagonal with the help of this autogenous pressure and temperature, and this crystal reorientation give rise to the formation of graphene quantum dots. The optical characterization, and of course, for a fluorophore, the stability is an important factor, and the quantum yield. So we have seen that the quantum yield of this product is 47%, and the stability is also there. We have done this lifetime, this time resolved PL's spectrum, and we have seen that it gives double exponential curve, giving the nanosecond approach, that 9 nanosecond, 10 nanosecond. So the photostability, how much it can go and be stable for the deep tissue imaging, that we have we have postulated in our paper. We have done the cytotoxicity with the cancer cells, with the normal cells, and even the hemotoxicity as well as the histology, because we claim that this is a biocompatible material. For biocompatibility, the material should be hemotoxic, cyto, and tissue compatible. So once it is done, then only that material can be used for biological applications. So we have done the bioimaging with the Hg2 cells. And this paper was again featured as a cover page in Green Chemistry of issue 16. And then with this material, we have formed this Janus-like structure. This I have given the artifacts, the morphological characterization. Did I told you about the basketry kegom? So this is the kegom basketry. How do I know it is a kegom? How do I say that this is a kegom structure? From the morphological studies. As I told you, interlaced equilateral triangles that can be seen from the HR10 images and the SAD pattern, which gives the HKL planes depicting the formation of kegom. Then the morphological characterization of the Janus at higher concentration. I'm not going into the details of it because of the time crunch, the elemental mapping and X-ray. Yes, kegom is formed that we give with the help of the peak 14.6 degree 2 theta. Now, how do I do it? Because people have reported about kegom with the help of metal oxide, and they have seen the peak from 14 to 16 degree 2 theta. Similar sharp peak was obtained at 14.6 degree 2 theta, confirming the formation of kegom along with the SAD pattern and the HR10 images. So we have done the differential scanning electrometry. Professor Bohidar has helped me a lot to do the DSC and the rheology, because we need to know the storage modulus, the bulk modulus, the degree of crystallinity, and then the loss modulus and the mechanical strength, that how this mechanically they can be used. Is the filler giving rise to soft gels or strong gels, and how they can be utilized. So that has been done. Swelling kinetics, and then the drug loading and drug release profile has been studied. And we have labeled the drug with technetium, and we have seen in rabbit model, and that has been validated with the help of gamma scintigraphy. Since we are not restricted our work to the rat model, which Dr. Arun and Dr. Satinder Rajput has done to us, we have gone forward to take it to the rabbit model, and we have seen that how it is going for sustained release of drug with the help of kegom and Janus to the colon without getting spread, which normally colon for 5-fluorouracil, if you use as it is, then it is a neurotoxic drug and it gets spread over the brain, liver, kidneys, and it also affects the eyes. So these, in order to avoid that, for sustained release, we went with the help of this material, which are biocompatible, the kegom and the Janus. Among that, the kegom is a strengthening material, more powerful material compared to the Janus if you talk about the mechanical strength. But if you tell about the release and the swelling kinetics, the Janus is better. What does it mean? It means that graphene quantum dot at higher concentration give rise to the soft gels. These soft gels are having such good applications in biomedical arena. So this work was, again, I'm not able to go into the details, was published in ACS Nano. I again take the privilege that this work has been done all from Amity University as corresponding author, and my students, from which I'm a chemist, I learned from them biology, has helped me to come out with this kind of publications. Then again, we have done with scarless healing of burn wounds, which I am not showing. 2nd degree burn wounds we have done. And we have said that, again, depending on the nanofiller, how we are going for the higher concentration, giving rise to a heptazine-based. It is a heterogeneous system, and how heterogeneous system is efficient for scarless healing of burn wounds, but homogeneous system is helpful for diabetic wound healing. So these are the results. And then again, some of the work, these are the 2nd degree burn wound healing. Yeah, I'm just stopping, sir. And this was published again in ACS Applied Polymer Materials. We have done a very good work which was published in Nano Today related to the coronavirus. And now I'm working with Sujata Sunil of ICGB to take this work forward. We have developed 9 different type of bioisosters using carbon quantum dots and triazole, using the click chemistry approach. And these materials are again biocompatible, and they are showing good trial result with the COVID strains. I'm still in the process of working on it, but since the molecules were developed and the basic results were shown, this work got published in Nano Today, and the methodology behind this, because it is very difficult for organic molecules to be established. We need to do the derivatives. We need to see the biocompatibility of the precursor, the middle one, as well as the derivatives before we go for the biomedical applications. And we have given the methodology of preparation of this, then proposed the inhibitory mechanism, which we have given in our paper, but it is yet to be supported by the final results by Sujata Sunil, who is working on these aspects. We are also working with algal nanobionics, which I have introduced recently, formation of bioactive compounds, lutein, as well as the formation of lipid, and how the graphitic nitrogen and pyridine nitrogen has a role in giving out the bioactive component, and the mechanism behind their formation has been depicted, and that gives rise to the lutein formation, and lutein is very good for eyes treatment. So this work again was published in ACS Sustainable Chemistry, and the recent work which I want to show you, I have come up with, if I get time next time to work on my paper, then I would tell that this is the most interesting work which I got, the injectable hydrogel with the help of click chemistry. And we are trying to work with this with Yogesh Verma, that how it can help in the hemostasis. So these are all the works, again, I am not showing. And the last one, and not the least, the stem cells differentiation. I was very inspired by Professor Das' department, Dr. Shubhrajit, Dr. Manoj, Dr. Vineet. Then I thought, let us work with mesenchyma stem cells. So mesenchyma stem cells differentiation to chondrocytes. It is, yes, why not pluripotent? Why mesenchyma stem cells? Why not other hematopoietic stem cells? The answer to this question is the mesenchyma stem cells has the ability to work with the scaffolds. It doesn't have HC2. It has, but in a very lower concentration. So the rejection of the scaffolds is less. So the results will be very good. And we got the chondrocytes, the cartilage, without the help. What is the USB of this project? Without the help of growth factors and inducers. People normally use growth factors and inducers for going to a particular lineage. But the beauty of these compounds, carbon quantum dots, biogenic carbon quantum dots, that they help in taking the particular lineage without the help of growth factors and inducers and giving rise to chondrocytes. And that we have given the mechanism with the help of Western blot and other experiments, that it is the MAP kinase pathway which is helping the carbon quantum dots to help this mesenchyma stem cells. We have gone to the gene level as well with Dr. Sujata Mohanty of AIMS, and we have done this work. And again, I take the pleasure to tell you that this work has been done by my students, Misbah, Abhayam Selvam, Om Narayan Selvam, who is a student of Amity and who got the opportunity to work in UC Berkeley. What more? Even in IITs and IIC, we don't get this kind of students who will go with this, because he has published 5 papers, filed 2 patents. So Abhay got the opportunity, and Nisba got the opportunity to work with Pennsylvania. Radhika and Om, they are working. Om will be flying to US. He's working with me for last 2 years in my UGC GE project.
Thank you, Mona. And now let's give a big clap to Mona. Mona Lisa.
So that's all about. And the profile of my faculties, I'm very less educated. If you compare the faculties of my department, they are all Ramalunga, Ramanujan, and they are doing great work. 2 more will be joining very soon. But yes, I have the desire, passion, dedication, and perseverance to work with them, to speak with them, to discuss with them, learn from them, and go ahead. And that's all about. Thank you.
Fantastic. Let's give yet another big clap for this outstanding scientist, Dr. Mona Lisa Mukherjee. And Mona, we are proud of you. Amity is blessed to have such brilliant faculties whose passion is only research and innovation. Yeah. And you are also a great motivator for your students, scholars, the way you work with them, and you also network with other institutions. Amazing, because you are not alone, not only within the Amity group, but you are networking with the AIMS, you are networking with IGIB, many other institutions. So we are really proud of you, and also John Moses in click chemistry. And your finding is very, very important because you are bringing out a solution for diabetic wound healing. Yes, we would like to see the deliverable coming out soon. And also the injectable hydrogels is yet another very important lead which you have. But now you start translating them into deliverable. What all need to be
And
done?
also the stem cells, sir.
Stem cells. So these are
Yeah.
the three
Yeah.
excellent leads.
Need to learn a lot of biology from Dr. Hridesh,
Yeah.
Dr.
Hridesh is here.
Vineet, Dr.
Now
Manoj.
let us hear the expert. I would like to invite Dr. Himadri Bohidar. He is from Jawaharlal Nehru University School of Physical Sciences. Very, very senior professor. Professor, can we see you on the screen?
Yeah, I do see you. Thank you.
He is PhD in physics from Indian Institute of Technology, Delhi, Marie Curie postdoc, University in France, visiting scientist of University of Oslo, Norway, visiting professor, Indiana University, USA, visiting professor in New South Wales, Australia. Then also, his specialization is molecular physics, soft matter, nanomaterials, composites, mostly interested in the nano-bio interface. This is his field. And his skills are in hydrogen. That's the area which he has worked. Rheology is another field. Biomaterials. So we have invited the right person to look at this and guide us. And what are his impressions on this presentation, on the work that is being done by Dr. Mona Lisa? And how do we join hands with you? And maybe that you are already partnering with her, collaborating with her. Over to you, Dr. Himadri.
Yeah, thank you, sir, for this nice welcome. Well, I am a partner to Dr. Mona Lisa for a long period, and I am quite familiar with what they do at their institute and keep pace with their research work and also read their papers very regularly. So, see, my overall impression about the activities that our group is pursuing at MET is there's one thing that is really remarkable. She has a specialization in the area of biochemistry and biomedical engineering and all that through her training. But during the course of research over the last couple of years, she has developed into a wonderful scientist where she touches upon topics deep in physics, biophysics, polymer science, nanoscience, and she has created a wonderful blend of the subjects, and that has helped her to take the research of this nanoscience domain into newer heights, and we can see it from her research publications and the kind of work she is doing. These are really admirable and wonderful work. I keep telling her all the time whenever we meet and we have interaction, and really world-class work. And what another thing which is remarkable about her work is she mostly does green chemistry, number one, and two, low carbon footprint. So these are the key words of the day, and this is where she makes her presence felt in the scientific community of the world internationally. And I'm very glad to see that the work she has done on the gel helixes, ice helix formation in the quantum confinements of a polymeric structure is going to yield formulation which can possibly help us in solving diabetic wound healing problems. So overall, my impression is quite positive. I wish her all the best. And and of course, the last thing, which is not the least, is she is able to collaborate with a lot of people, a lot of people in a lot of institutions, which is really there. So I wish her all the best.
No, including you, I said, Dr.
Including I am a very minor partner, but in addition to me, there are many others who collaborate with her very actively.
Thank you.
Thank you very much.
So, Mona, these are the words of inspiration for you and your team, and your publications are outstanding. ACS Nano and Nano Today and Advanced Materials, Material Horizon. These are all top journals, RCS, Royal Society of Chemistry journals.
Right.
Top. So we wish you many, many more successes. You have worked with a Nobel laureate. So we want a Nobel laureate now. So, Mona, you should now aim dream.
got the opportunity to talk.
Yeah, no,
with the
I
help of
think you
John.
should now aim at Patnagar Award, and also you should aim at Nobel Prize. So all this is possible. They're not somebody who came from
I've
elsewhere.
already passed.
So let's wish her with a clapping. Thank you so much, Mona, for this wonderful work. Keep continuing
Hopefully,
this
sir,
work.
I tell you, I think I will be doing very good work because I have come in close proximity to work with renowned Bhatnagar awardee and Dr. Malika. So I hope we will do a very good work in geography soon. Isn't it, sir?
Outstanding.
We hope.
Thank you. Wish you all the best. Blessings.
All the best.
Thank you. Thank you, Dr. Himadri.
Thank you. Thank you. Thank you.
Now,
Thank you,
we move on to
Dr.
the next
Himadri, sir.
presentation by Dr. Durgesh K. Tripathi. Again, a young, budding, as well as a very accomplished scientist who is in top 2% in the Stanford survey. When they carried out, we have about 28 of these from Amity Group in top 2% scientists from across the world in different domains. So now he is one of them in the field of agricultural science, plant science, and he is also in top 1% survey, top 1% researcher around the globe in 2021 by the Clarivate analysis, Web of Science. And this person has done outstanding work, and he is PhD from Department of Botany, University of Allahabad, that is in 2012. His research areas are genomics, molecular breeding, and plant nanobiology, plant nutrition, growth and development mechanisms of stress, adaptation, stress and adaptation, metals and metalloids, stress management in plants, and silicon and hormonal crosstalk under heavy metal and nanoparticle stress in plants, many, many more. So we want to listen to Tripathi. Tripathi, he is now in the Amity Institute of Organic Agriculture, Amity University, Uttar Pradesh, Noida, and he is going to decode the silicon potentials in plants. Over to you, Dr. Tripathi.
Thank you so much, sir. So, before begin my presentation, first of all, I would like to thank Honorable Founder President, sir, Chancellor, sir, Honorable Vice Chancellor, ma'am, Dr. Silva Murthy, sir, and Professor B.C. Das, sir, and other dignitaries for providing me such a wonderful platform to present my research finding. So, the topic of my research is decoding the silicon potential in plants. This is
Take about 20 minutes for presentation,
right.
10 minutes for discussion.
Okay,
20 minutes.
Okay, sir. Thank you. This is a beautiful campus. So I would summarize my presentation under the following 4 objectives, major objectives. Like first one is identification of silicon deposition pattern in crops. There are some sub-objectives, and the second one is decode the mechanism of silicon-induced deliberation of abiotic stress in plants. Silicon or silicon nanoparticle, which one is the best form for the future agriculture? And the fourth and last one is concept of biogenic silicon nanofertilizer for crop growth and abiotic stress tolerance. So I would like to first introduce why I have chosen the silicon and what is the meaning of silicon. Silicon is the second most abundant element in the earth crust after oxygen, yet it was not selected as the building blocks of the life on earth by the nature, probably due to its undesirable chemical interactions. But the presence of silica in different life forms such as diatom, silica deposition, or silicified cell, generally known as phytolith. Phyto means plant and lith means stones. So it may, in many terrestrial plants, highlight the nature's generosity in keeping silicon to play a role in life on Earth. Silicon can be a potential contender as the alternative to carbon for the elemental basis of life in the environment, with very different biochemistry from what the Earth has, despite more than 100 years of work on the silicon as essential or not essential nutrient, which is a major part of the discussion. The detailed basis for its presence in plant life, mechanism induced or altered by silicon in plant, and other possible implications were not systematically addressed. Therefore, I begin my research with following objective, which I have already so you, all of you, in my first slide. So this is the first objective, identification of silicon deposition pattern in crop. Means if plant is accumulated silicon, so in which form it deposited into the plant cell. So this was my first objective. These are the sub-objective: deposition, distribution, and concentration. Whether it is deposited in the edible parts, because we are consuming so much of food product from agriculture crops. So I have also I have also analyzed whether it is also deposited in the edible parts or not. And the last one is impact of silicon against the abiotic stress, whether it is have any role in the abiotic stress or not. So first, I have selected two sorghum species, and just with the help of microscopy, I have analyzed that the silica plant deposited the silicon in different plant cells, very unique structure of the plant cells, generally known as bulliform cell, prickle hairs, dumbbell shapes, and other very unique form of the cell. Another study, I have also selected the wheat plant, and I have analyzed the silica deposition pattern with the help of microscopy as well as the spectroscopy in leaf, stem, racula, and own, and have analyzed that in which form it is highly deposited. And these two papers published in Proceeding of National Academy of Sciences and Agriculture Research. Another study, a very common grass, Cynodon dactylon, which we generally utilize to beautify our garden. So in this study, I have selected the Cynodon dactylon and have analyzed the upper surface of the leaf and the lower surface of leaf, and utilized the laser spectroscopy analysis and microscopy analysis, and found the similar kind of result, that silica deposited in very beautiful, unique plant cell. This study has been published in Food Biophysics, very reputed journal. Now, another study, I have selected the plant Cucumis sativus. This is the common name of kheera, which we generally utilize in our food. Kheera, and I have analyzed that whether silica is deposited in different parts of the food product. It found that silica is also deposited in Cucumis sativus. And this study has been published in C Biotech. Now we have a question: why plant accumulate the silicon and deposit it in different plant cells, whether it has any significance or not. So I have just applied the exogenous form of silicon in the form of sodium silicate against the abiotic stress, because we are all over the world is suffering from different problems of abiotic stress. So that's why I have selected sodium silicate and have started to giving exogenous application against the chromium toxicity in the wheat seedling, and with the help of laser spectroscopy analysis, microscopy, and biochemical analysis, I have found that silica is very effective against the abiotic stress, means chromium toxicity. It has capacity to control the chromium toxicity in the wheat plot. Same kind of study I have analyzed with the help of whether the biochemical parameters, biochemical means any metabolic process are involved or not. So I have also applied the silicon against the chromium toxicity in rice seedling, and found similar kind of result, that silica is also very effective to control the chromium toxicity in rice seedling. These both paper has been published. This one is published in Ecotoxicology Environmental Safety, very important journal, and this has published in Acta Physiologia Plantarum. Next, I have selected one of the major abiotic stress, that is herbicide, pesticide toxicity, because we have utilized pesticides from very long time, and now our soil is very much contaminated. So now we have some kind of solution. So I have selected butachlor, which is very toxic and generally utilized in the rice field. So I have applied the silicon against the butachlor toxicity, and found that silica is also very effective to control the pesticide toxicity. These are some result that how silicon have capacity to manage the toxic impact of pesticide, and this paper has been published in Nature Scientific Reports. These are some results. Now, what are the conclusion and societal benefit of my first objective? So these studies highlight the presence of silica deposition in important agriculture crops and imply their probable significance. Also, silicon was found to be potential alleviator of abiotic stress in crop that can enhance crop productivity by minimizing the stress-induced losses. These research projects that silicon fertilizer of the soil by the farmers can help in increasing the crop yield even under favorable growing condition and can bring financial benefit to the farmers. So this is overall the conclusion and societal benefit of my first objective. Now we have at that time I have a major question that if silica is effective to control the abiotic stress and plant can easily accumulate the silicon, then what is the mechanism, which mechanism is work behind, or which mechanism support to the silicon to protect the plants? So in this objective, I have started working on decode the mechanism of silicon in the alleviation of abiotic stress in plants. And we all know that silica, phytohormones and signaling molecules, they are a very good player to regulate overall plant life. That's why I have just analyzed that whether silicon has any relationship with the phytohormones as well as the signaling molecule like nitric oxide and hydrogen sulfide. I have also selected two hormones, major hormones, indolestic acid, generally known as the auxin, and salicylic acid, and to find out what is the mechanism working and what is the system support to the silicon. So I have applied silicon with the help of first paper, with the help of indole acetic acid against the arsenic toxicity in the rice plant. This study has been published a very international reputed plant science journal, that is Journal of Experimental Botany. And so I have analyzed that silica indole acetic acid have a very good interaction with the silicon. And when we applied the silicon, indole acetic acid increase the advantageous root formation to dissolve the toxicity of arsenic. And this was a very good finding of our research group, and this has published in Journal of Experimental Botany. These are the molecular studies, the result of molecular studies, as well as this is the mechanism which we have decoded. What is the interaction of silicon with the indole acetic acid? Another study I have selected the salicylic acid, as I have earlier included that we have selected two hormones. One is indole acetic acid and second one is salicylic acid. So I have I just studied what is the interaction of whether only silicon and indole acetic have a relationship, whether it has a relationship with some other phytohormone. So we have analyzed the salicylic acid, its impact of salicylic acid along with the silicon, and found that it is very effective against the toxicity of the cadmium in maize seedling. And this study has published in Journal of Plant Growth Regulation, a very reputed journal. This is the mechanism which we have isolated, and this has already been published in very good journal. Next, we have in two papers, in two previous studies, we have analyzed what is the relationship between the phytohormones as well as the silicon. Now, the second sub-objective was what is the relationship between signaling molecule, another very good player for the plant life, signaling molecule and the silicon. So I have selected the nitric oxide to decode what is the relationship of the silicon and nitric oxide, and we have combinedly applied against the toxicity of the copper in the mung bean, and found that silica have a very good relationship again with the signaling molecules. It means the internal integrity, means internal metabolic system, including phytohormones and the nitric oxide. They are supporting to the silicon to control the toxicity of any abiotic stress, including copper, including pesticide at all. So this study has published in Plant Physiology and Biochemistry, and we have decoded a mechanism that silicon has a very good relationship along with the signaling molecule. These are some results we have published. Next, this study has published in this year a very reputed journal, Plants in Physiology, published by the Oxford University Press. And in this study, this is the project work of SERB funded project work. Silicon palliates the toxicity through the formation of root hairs in the rice mediated by GSH and IA. During the first study, when I have first time applied the silicon against the chromium toxicity, I have observed that under the chromium toxicity, plants increase the root hairs. So in this study, we have just analyzed why the chromium is increasing the root hair formation, and whether it has any role or not when we have applied the silicon, and found that silicon increase the root hair formation so that plant can easily dissolve the toxicity of chromium. And this study has been published in Plant Cell Physiology. This is the mechanism which we have decoded. Now come to the conclusion and societal benefit of my second objective. So these research studies were driven by the desire to better understand the mechanism involved in silicon-assisted growth promotion and stress tolerance in plants. We have on the conclusion that the internal plant metabolic system is very much supporting towards the silicon application. Silicon was observed to regulate a complex internal network comprising of various signaling molecules and phytohormones to achieve the desirable beneficial impact of the plant. Delineating these details can help in understanding the possible implication of silicon as a fertilizing agent on different crop as a response vary with the plant species and growth conditions. So these are two my major objectives and their conclusion and societal benefit. Now the third objective, which is very important and very interesting for me as well, because we all know at present living in a nano era, we are utilizing so much of things which is made by the nanotechnology. So I have also a question whether if we apply the silicon nanoparticle in place of the silicon, so what will be the impact? So I have started a synthesis of the silicon with the help of sodium silicate in the form of nanoparticle. And I have also a question at that time that which is the best form for the future agriculture. We will be facing so much of problem 2050 all over the world for the food hunger. So keeping in this fact in my mind, I have started synthesize silicon into the silicon nanoform. These are my two sub-objectives: impact of silica nanoparticle in plant and abiotic stress. Whether I have synthesized the silicon nanoparticle, but the question is whether it is effective or not. Because silicon is effective, that's why I have a framework, I have a background that silicon nanoparticle may be effective. Second is comparison of silicon and silicon nanoparticle. Which form is the best? So these were my two sub-objectives. So I have started working first on the silicon nanoparticle. Again, in a Python sativum seedling against the chromium toxicity, to know whether silicon nanoparticle are effective or not. And I have found that silicon nanoparticle is very effective, and it can control again the silica. It can control the chromium toxicity. This study is published in Plant Cell Physiology, a very plant science reputed journal. Again, I have another question that whether the silica nanoparticle have the same kind of relationship with the phytohormones or not, because silica have a good relationship with the phytohormone and signaling molecule. So if I am going to convert into the silica nanoparticle, whether these have the silica nanoparticle have the same kind of relationship, because because any exogenous element which we applied internal plant system should be supportive, then we can achieve our objective. So I have analyzed that synergistic action of silicon nanoparticle and indole acetic acid in elevation of chromium toxicity in Raja sativa seedling, and found that indole acetic acid again support to the nanoform of the silicon, which is very exciting result and published in Journal of Biotechnology. These are the mechanism that how the silicon nanoparticle and indole acetic acid interact and how they can control to the toxicity of the chromium toxicity. Now the second sub-objective, which was my major objective. Which form is the best, silica nanoparticle or the silicon? And I have started working against the arsenate toxicity in the maize cultivars. So I have found that silica nanoparticle more efficiently alleviates the arsenate toxicity than the silicon in maize cultivars and hybrid, differing in arsenate tolerance. Means what hypothesis I have made at that time, that if I will apply, I'm going to apply the silica nanoform. So whether it is more effective against the silicon or not, I have found that silica nanoparticle is very effective, and it can control the arsenic toxicity, which has the major abiotic stress component. And this study has been published in Frontier of Environmental Science. Again, I have selected another abiotic stress, ultraviolet radiation, to cross check whether it is only effective against the heavy metal toxicity or the metalloid toxicity, or it have capacity to control some other abiotic stress. And I have found that silicon nanoparticle is similarly, as my previous study, is very effective against the ultraviolet radiation, and it can control the ultraviolet radiation in the wheat seedling, and silica nanoform is again very effective. So now come to the conclusion and the societal benefit of my third objective of my research. I have found that silica in its nanoform was found to be more effective than its bulk form in promoting growth under normal as well as the stress condition in different plants due to its large surface volume ratio and easy availability to plants for uptake. Therefore, silicon nanofertilizer can be better option for the farmers to receive more benefits in comparison with the silicon supplementation to enhance crop productivity and minimizing the losses of the crop to environmental stresses. Silicon nanofertilizer will make silicon ion easily accessible to plants, and therefore the amount required will be less than silica bulk form to gain the equivalent beneficial impact on the crop. So this was my conclusion and societal benefit of my third objective. Now come to the final and last objective, concept of biogenic silica nanofertilizer for crop growth and abiotic stress tolerance. After these three objective, analyzing after three objective from last 10 years, I have a question in mind that if silica nanoparticle are very effective for the plant system, and it can enhance the growth and productivity even under the abiotic stress condition, then why we are utilizing the chemical form of the silicon? As I have earlier reported in my very first objective, that silica plant accumulate the silicon in the plants. So why I should not isolate from activated form of silicon from agriculture byproduct? Because every year farmers they are burning the agriculture byproduct, which is a major problem in Noida and New Delhi area. So I have prepared a proposal and submit to the SERB, and they have granted me, and I have started working on the concept of biogenic silica nanofertilizer for crop growth and abiotic stress tolerance. Biogenic silica nanofertilizer, I am the first person with all blessings of my teachers and the dignitaries sitting over here. I have the first person who have given the term BSINP, biogenic silica nanofertilizer, in the field of silicon research. Biogenic silica, so these are the two sub-objectives: biogenic silica nanofertilizer impacts, comparison among the SINP and biogenic silica. This is the second major question before me, that if I am going to utilize the biogenic silica nanofertilizer, which form is the best? Silica nanoparticle, which I have synthesized from the sodium silicate, silicon alone, or biogenic silica nanofertilizer, which I have prepared from the agriculture byproduct. So this was my major objective, and I have started working under this objective, and this research is ongoing, and I have found very interesting result. I'm going to submit a patent along with Dr. Sangeeta Pandey madam, because I have also taken her support in. I have some of her support in her lab. So I'm going to submit a patent, and this was so I have isolated the silicon from the rice husk, and then characterized systematically and found that biogenic silica nanofertilizer, the fourth one, is very effective against the chromium toxicity And it will not only control the chromium toxicity, but it also alone enhance the root length, shoot length, and it have capacity to improve the yield. So this is overall my result of the 4 objective. Now the conclusion and societal benefit of my 4th objective is the impact of biosilica nanofertilizer is more prominent on plant growth. Biosilica nanoparticle help the plant to cope better with the stressful condition in comparison with the SINPR silicon. The probable reason for the same could be the derivation of biosilica nanoparticle from the plant bioproduct. Only that might attribute for the enhanced biocompatibility. Biosilica can be of great importance to the farmers as they offer a more crop-friendly and environmental-friendly sustainable alternative to chemical fertilizer. The serious issue of burning the crop residues in major food-producing state of North India, Haryana and Punjab can also be resolved by using them for synthesis of biogenic silica nanoparticles. Now, what is the overall societal impact of my study, which I have continuing from last 10 to 12 years? The primary issue faced by the modern day agriculture is the toxication of the environment by the prolonged use of chemical fertilizer to boost the productivity of the plants. Silicon and its nanoform, especially biogenic silica nanoparticles, offer a sustainable alternative to these chemical fertilizer. This approach of combining agriculture byproduct with the most promising technology of present day, that is nanotechnology, can revolutionize the agriculture sector. The beneficial impact of silicon can be seen in the overall food quality obtained from the plants. Availability several plant-based silica products for human consumption also assist this notion. Silica-based mechanism of enhanced plant productivity and stress tolerance can be employed further to develop stress-resistant variety of the important crop. This can solve the problem of increasing food hunger worldwide by increasing the food production. So this was my conclusion and overall societal benefit of my last objective. I have completed my overall 4 objectives. These are some outstanding publication which I have published along with my PhD student, Nidhi Khandol. This very renowned journal, Training Plant Science, Impact Factor 22.1. So all these papers I have published from our esteemed campus, Amity University campus. So Training Plant Science, the first one. Second one, again, Training Plant Science, 22.1 impact factor. Arsenate, the empire of arsenate perception and responses in plants. Training Plant Science, nano carrier spray, a non-transgenic approach for the crop engineering. Another outstanding publication along with my PhD students. Another paper published in Plant Cell Physiology in Oxford University Press in this year, in the last year, 22, along with my PhD student. Nanoparticle as potential hallmark or drought stress tolerance in plants, which I have published with Professor Mukesh Jain, Professor and Dean, Jawaharlal Nehru University, New Delhi, in Physiology of Plantarum. These are some of my other publications which have been published in different international world-renowned journals like Plant Science, Training Plant Science, Nature Scientific Report, Hazardous Material, Critical Reviews in Biotechnology, Journal of Experimental Botany, Physiology of Plantarum, and many more. What is the leadership of my leadership in the silicon field at the global platform? I would like to just explain that I have edited one special issue, Journal of Experimental Botany, which is very renowned journal in the plant science, as a lead guest editor in a Journal of Experimental Botany. Another special issue I have initiated from our Amity University. All these work has been completed from our esteemed Amity University campus. Plant Physiology and Biochemistry. These are some books we have published with Academic Press, Springer, elsewhere. Books published, and this is over my profile. Total number of publication 205. Total number of citation is 8,665. 21 books published. I'm associate editor with the grace of God and all your blessings. I have associate editor in different 12 international journals, very high impact factor journals. I have recognized as a top 1% scientist by Web of Science, Tony B. Academic Award, USA, Washington, D.C. Top 2% from 2020 to 2022, regularly. Science Communication Award by Indian Science Congress Association, National Academy of Sciences Travel Award, and so some other awards. I'm also acting as a chief editor in a journal, Plant Nanobiology, which is published under the Science Direct. While elsewhere, I'm acting as a chief editor and trying to represent my esteemed Amity University.
Thank you. Thank you, Tripathi.
And I have also established some international collaborations, collaboration with some American countries. Professor Vasila Julba from Lawrence Berkeley University, Professor Walter Gassman from University of Missouri, Professor James Jose Parlata from Texas University. Some collaboration with the European countries, Professor Mark Vekolic, he is from Communist University, Gia Aguario from Luxembourg University, Vasilis Fotopoulos from Cyprus University, and Professor Alicidar R. Freni, world-renowned person, well-known, published 2,233 publication from Max Planck University. He's also my collaborator, and I have published one paper with him. Some are still ongoing. Ongoing project from SERB, CSIR, and MHRD. 2 projects is still ongoing, and some project, 4 projects I have completed, in which 3 Gyan project, 1 CSIR project, and 2 are still going. Recently, along with Dr. Sangeeta Pandey ma'am, I have received a grant to organize Karyasala from SERB, 5 lakh rupees. So this is the SERB funded research lab. I have established the crop nanobiology and molecular physiology lab. These are my students, and Dr. Sangeeta Pandey madam, or some international visitor they have visited this lab, which I have established with the support of our esteemed Amity University and SERB. I would like to acknowledge Amity University Uttar Pradesh Noida, Department of Science and Technology, DST, CRB, Council of Scientific and Industrial Research, Motilal Nehru National Institute of Technology, Allahabad, University of Allahabad, and Banaras University for continuous support me for my research work. Thank you so much, sir.
Fantastic. Let's give a big clap for him.
Dr.
Founder President, sir. Welcome, sir.
My appreciation to Dr. Durgesh. Wonderful. I am so impressed, so joyful, because such brilliant work what I have heard here. And I will also hear now our Dr. Rupesh Deshmukhji and also our Dr. Shivani Sharmaji, Shivesh Sharmaji, because this work is really so useful. The other day, Modi ji was very much propagating the nano fertilizer, and he felt pride of our country. So I think, Durgesh, I will meet you with Dr. Murthy separately also. I want to read you send me 2-3 of your papers, which I want to read, but I'm really, Dr. Murthy, you and your team, grateful for organizing such a wonderful. I was till now in other, but I was listening in between. I was attending both the programs. So I think wonderful. But we should, Dr. Murthy, engage ourselves more with Durgesh.
Right, sir.
Thank you so much, sir, for your kind blessings.
Amity is blessed.
It is a great pleasure for me, sir.
Great.
Thank you so much, sir.
Amity is blessed to have such brilliant, young, dynamic leaders in science and technology. Congratulations, Dr. Tripathi. Now let us hear the 2 experts. Dr. Rupesh Deshmukh. He is the professor at the Department of Biotechnology, Central University of Haryana.
Right.
And he is one of the outstanding scientists in the field of agriculture. PhD from in the field of agriculture biotechnology, postdoc. He has done 2 postdoc, one at the University Laval, Quebec, Canada, and the other one is in University of Missouri, USA. And he went back again to Laval, and his field, he has got many awards. Ramalinga Swami Fellow, he is a Ramalinga Swami Fellow, and Nasi Scopus Young Scientist Award, and Fellow of ISGPB. And also he is among the top 2% scientists published by Stanford survey both in 2021 and 22. He has 150 papers to his credit, and his specialization is plant genome. And he is also editor of Plus One, BMC, and many, many accomplishments. We are proud to have you, Dr. Deshmukh, with us. Can we have your views? And this person, young scientist of Amity University, is bubbling with energy, enthusiasm. We would like to hear your advice, guidance to him. Over to you.
Thank you very much, sir. And first of all, I want to thank founder, president, Dr. Chauhan, and also vice chancellor and all the leadership positions at MIT for such a wonderful series that is like Pathbreaking Scientific wonderful initiative from MIT. And I enjoyed listening both the other stuff was not from my area, but it was really wonderful to see the scientists at MIT doing such wonderful work. And I really impressed with the multidisciplinary expertise which MIT has. And I think that is the benefit or that is the advantage of working at MIT. And that reflects really very well with the work from Dr. Durgesh. And I am following him from last 10 years. And I just wanted to share one small incident, how I came to know about him. Because I did my PhD at Pusa campus, New Delhi. That time I was working on a plant genome. And then I shifted to Canada in University Laval. That lab was working with the silicon. And that's how I introduced to the new field like silicon biology. And that time, people were talking about the global leader in silicon biology, like Jiangpeng Ma from Okayama University in Japan, then Hudson, Lux Alexander, and many more names. And then people started talking about the book by Dr. Durgesh Tripathi. He wrote one book on silicon. And that was like a reference book for many scientists working in the silicon research. And that's how I started searching for a person. Like that time, I was like really, very, very proud to listen the Indian name in among international community in silicon biology. And then I searched for him, and then I went to meet him in Allahabad. And when I first time saw him, I was very surprised. He's so very young to become such a famous scientist in silicon biology. And still, it is very unbelievable for many people. Like he's a top 1% under top in a top 1% scientist. Like I am also in a top 2% of scientists, but there are more than 2,000 scientists in the top 2%. But if you consider the list of top 1% scientists, there are very few. And from plant science, I think only a couple of Indian names are there, like Dr. Durgesh Tripathi, Dr. Manoj Prasad, Dr. Raju Vashni. I think 3 or 4 names are from plant science. And this is such a huge achievement in such a young age. And I really admire him as a leader in silicon biology. And as I already shown his work in several different objectives. And I want to really highlight that each and every objective has a social impact. And if you now in today's situation, if you go in any agriculture shop, like seed shop or pesticide shop, you will find a silicon-based fertilizer. And that is the really outcome from the research done by Dr. Durgesh or many other leaders in silicon biology, like Dr. Richard Banerjee, Balaji, or Jiangpeng Ma. So he's among those who really bring this silicon fertilizer in the market. And these silicon fertilizers are more common in Indian market and African markets, because these are the countries which are more dependent on the agriculture, and they always want to explore the new sources of fertilizers which are more economically better options, as well as we also started caring about the environment. And his research is really very important when we are talking about the heavy metal stress, and also for the our new sustainability goal, that is the converting waste to wealth. That is like rice straw or rice husk, which is a big issue in North India. And if we are able to convert that into any product, or any way we can recycle that effectively, that will be a really very good societal impact. And already Dr. Durgesh Tripathi is doing very well in this area. He has already developed silicon nanoparticle from the rice husk. I think such initiatives from his group will really show up in the market with deliverable form of products. And I really congratulate him for such a wonderful work in silicon. Thank you very much, Dr. Durgesh, for your contribution in this field.
Thank you.
I am also following almost all of his publications. Like he are published in really very well reputed journals like JXB, then PCP, then Trends. These are all very reputed journals in plant science. And I can count like 3 of his findings, which are really changed this area of silicon biology. Many of you are not from, I guess, not many of you may are not from the plant science or not aware of this core area of silicon biology. There are very few people in India who are working with the silicon, and globally also not many people are in this core area. But this is really impactful area. And Dr. Durgesh finding of how silicon regulate the plant hormone, that was really a path-breaking discovery. Because the work on silicon was started almost 100 years ago in Japan, but everyone was showing that the silicon is beneficial for the plant. And that's why now we have several silicon-based fertilizers. But there was a knowledge gap that no one was knowing that how silicon exactly benefits the plant. And when Dr. Durgesh started publishing his papers on how silicon regulate the plant hormone, then the entire picture becoming more clear. And then the second of his path-breaking discovery, like the silicon role in signaling molecules, like nitric oxide. This is relatively very, very new area. I think only his lab is working on that area, that role of silicon in signaling molecules. And I really appreciate this work. And we are very closely following this work. Like his findings are very important for us to translate this research in a different product. Because my lab is basically working with the plant genomics, and I am more to the plant breeding background. So whenever the paper comes from Dr. Durgesh lab, we try to identify the genes and to incorporate those genes in different breeding lines. And the third area he started, that is the silicon nanoparticle. I think in India, this is very important because now we started using silicon fertilizer, but many of the Indian crops, they are not able to uptake silicon, like chickpea or brassica. Those crops are unable to uptake silicon in any form. So these silicon nanoparticles are very efficient for such crops where there is no gene to uptake silicon from the soil. And I think Dr. Durgesh's discovery will be very helpful in those crops specifically. So I just want to ask one question to Dr. Durgesh. So if you want to take this your experiment or your results to convert into deliverable form of product, so what you will recommend, like going to a biogenic silicon as a fertilizer or silicon nanoparticle based fertilizer or biogenic silicon nanoparticle as a fertilizer? Because now we have like different regulation to take this nano technological product in the market. So whether you are already started working in this direction, or what is your opinion?
So thank you so much for very wonderful question, because it is totally connected with my research work. Whatever I have analyzed during my entire of research work, I have found that silica, the nano forms of silica is very effective, right? But the maximum people, they are synthesize the silicon with the application of chemical materials, right? So I have isolated the silica from the agriculture byproduct, which I have already explained in my presentation, that every year farmers they are burning the agriculture byproduct because like rice husk, it cannot be utilized in any other form. It cannot consume by the cattle because rice husk generally utilized as a fuel, as a burning material, right? So if we just isolate the activated form of the silicon, because silicon is deposited in the soil in the form of rocks, right? Plants, they convert unactivated form of silicon into the activated form, and then they deposit it into the plant cell. If we can isolate the activated form of silicon from the agricultural byproduct like rice husk, bamboo, and so other many plants, they are recognized as a highly silicon accumulated. and then we can convert into the biogenic silica nanofertilizer, and then we can recycle into the agriculture field. It will be more effective. And this is my major concept. And this is the overall result of the SERB funded research project, which is still ongoing. And I have a very exciting result. So I would like to suggest that we should utilize the silica biogenic silica nanofertilizer instead of silicon nanoparticle, which has synthesized by the utilization of the chemicals.
I think this will also help to address the stubble burning issue.
Yeah,
That's right.
Every year we talk about this in December, January, and then again forget about this issue for the 1 year.
yes.
So I think this will be really helpful to address that one.
We can resolve the issue of this agriculture consumption of the agriculture byproduct, because every year we are New Delhi and Noida and adjoining area, they are facing so much of environmental pollution.
Yeah. Durgesh, so
Yes.
he has given a mandate to you
Yes.
that now you should look at the product.
Yeah.
Product
Yes.
deliverable.
Yes.
Now I would like to have your roadmap, how soon
Yes.
you can convert the scientific lead, knowledge, and wonderful research what you have done in Amity University. How would you make it as a wealth now? You can even think of a startup on your
Yes.
name, which founder wants. He wants 25 startups. So you have a product now, silicon-based fertilizer, nano
Yes.
fertilizer.
Yes.
So please start thinking in that direction, so that now convert this Saraswati into Lakshmi.
Right.
Now convert this knowledge into wealth. So
Right.
that is the mandate Drupesh also has given. Founder President is keen. Founder President, am I right? Just please unmute the mic. Your mic is muted.
Yes, you have the power to read my brain. This was my next sentence, that Durgesh and all other three, my great scientists, make their own company, because this company has many fold benefits, and also take in this company our partner, Mr. Rupesh, because they can be the partner in the company. And Chakravarti, Neeraj Sharma ji, you take up this job from the war room that they make, because I have told to that Satish Reddy ji that 25 companies we'll be making within 12 months after his visit when Satish Reddy ji came, because I am so convinced. I always say the earning what my faculty member has or anywhere else is not sufficient. Why should they not earn a lot? I have been an industrialist. Now change in academics. If these people will go industry, they will earn 4 times. But why should they not earn 4 times being with me, being in academic institution? So I think I have really not understanding that these brilliant people are getting less, because in a private self-funded or government, one cannot pay unending. But I can make them through patents, because we give them 20% on the commercialization. So I want to pursue with you people to be academically, scientifically innovative, brilliant, but also not less than anybody in the industry. So Lakshmati, I think all these 4 my scientists should immediately make a company. And secondly, while I was listening this, I rang up our chancellor, who is the chairman of the governing council, that I want to propose to Dr. Murthy, Dr. B.C. Das, Chakravarti and Neeraj Sharma, that we offer to all these 9 experts, I've not seen if all are there, we should offer them the honorary professorship. The honor from this, because I'm so convinced to listen them. So I think we should offer them the honorary professorship, and we should pursue with them to make their own company with the partners, those who are experts.
Thank you, Founder President.
Thank you so much, sir. Thank you so much for your kind blessings and guidance.
See, what a tribute to your work from Founder President.
Yes.
He wants to make you a billionaire. So immediately jump on this idea.
It's always my pleasure, sir.
Thank you so much.
Compliments
to you. Let's
Thank you
listen
so
to
much
the
for your blessings.
next expert, Sir Shivesh Sharma. Yeah, yes.
When I came from West Germany in 1994, people said, we have been at your place, politician. You can be the richest billionaire in India and very famous. I told, I don't want. I want to make millions of billionaires. This tactics I know, because if you have done anything yourself, very easy for me. Anybody should come to me. I want to be a billionaire. If I don't succeed, it's my failure. I tell it openly on record. openly. Thank you, sir.
Success by compulsion. That is your philosophy. Success by compulsion. Now we have another Professor Shivesh Sharma, another outstanding scientist of our country in the field of biotechnology, agricultural biotechnology. He is from the Motilal Nehru National Institute of Technology, Allahabad, and PhD in microbiology and environmental biotechnology, plant stress physiology. He has supervised 14 PhD students, scholars, filed 6 patents. One of the patent is granted, and he has transferred 2 technologies to the industry. And one of them is that low-cost portable UVC sanitization cabinet. And so this he has already delivered. So we are very happy to have you, Professor Sharma. We like to listen from you. What is the dream for our Durgesh? What is the dream you have for him? Over to you.
Good evening. I hope I am audible to all of you.
Yes, perfect.
Okay, so at the outset, I would like to thank MIT University for having me here as an expert in this path-breaking scientific achievement session. And I want to congratulate the founder president first, organizers, especially Dr. Silvamurthy and entire administration for such a nice and innovative initiative. So I must say that the presentation on decoding the silicon potentials in plants, which Durgesh just made, it was really very precise, but yet it was very descriptive. And I know Dr. Durgesh, and he has been working in the area of silicon biology and silicon research, and I am aware about his scientific contributions in this particular area. And I must admit that his presentation today showcased his experience and totally justifies his selection among top 1% scientists across the globe. It's really a matter of great pride for me because he has worked in my lab as a scientist, as a PDF scholar, and it's really a matter of great satisfaction as well as pride to hear such a appreciation from the founder president himself. And for the better understanding and clarity of the audience, I just would like to ask Durgesh some very important point. Because most of the audience who are present here might not be aware about this particular research, that silica is deposited in the edible parts of the plants. And as we know that we consume the food products which contain silicon, so please let us know whether silicon has any beneficial or toxic role in human health, because this question holds great significance with respect to the startup.
Yes, sir. Thank you so much for this question, sir. As I'm totally working on the plant science, but I have go through the very wide literature that before selection of the Cucumis sativus, that is the local name of the Kira, and I found that silica is deposited in the Kira, and we generally utilize as in our food. It's a part of our food. So I have go through the wide literature and found that when we consume silicon in the biogenic form, like when we consume any agriculture by any food, like it has also been reported in apple, in Kira, and several other food products. So when we consume a biogenic silica, it make a very thin layer in the arteries of our heart, our heart. It will help for the movement of our blood, right, sir? So as I have not studied yet, because I'm a purely plant sciences, but I have also I was very curious about why plant absorb silicon in the food products. So I have go through the wide literature and found that biogenic silica made a very thin layer in our heart, arteries of our heart, which help in the movement of the silicon. Which help in the movement of our blood. But when we consume the silicon, through our nose, means exogenous silicon, which is not biogenic silica, it is very harmful and it can create the cancerous disease in our body. So it is harmful when we consume the silicon as a dust or as an other and in other form. But when we consume the silicon in the form in the form of biogenic silica, it is very helpful and for our health.
Very good.
Okay.
Well, Jane.
Yes, sir. So just I would like to suggest one thing, Durgesh, to expand the horizon of your research. By working on new dimensions like nano encapsulation of fertilizers or nano-based nucleic acid delivery, since you just showed one paper on trends in plant science.
Yes.
So you already have started working, but still you can work on in this particular area, and definitely it can bring a new revolution as far as agriculture sector is concerned. So on this note, I once again congratulate you first for your presentation, and I wish you all the good luck in your future endeavors. Thank you.
Thank you so much, sir. Thank you. So much.
Professor
Thank you,
Sharma ji, already founder president has given an invitation for you to be an honorary professor. So we would send a formal note to you.
Thank you so much. Thank you. Thank you so much. Thank you.
It's an honor for us. Such an experienced person. You can guide him, mentor him, motivate him. And thank you so much for your very valuable comments. Safety of the nano fertilizer, which will get into the edible part of the plants.
Yes.
How do we take care of it? Very important. Now, can I have Dr. Nutan Kaushik? How you feel that now having heard both the experts, now how would you take it forward in terms of deliverable, the product, and also registration as a startup company? So we like to hear DG, Director General of the Amity Food and Agricultural Foundation.
Sir, Durgesh, very good presentation. I'm really happy to see this, all this, what has been presented for your past 10 years experiences, and particularly what you have done at Amity University. I would suggest that since most of the work has been done at the seedling stage, so now you should go for the field trials later on in the startup. Of course, this biogenic extraction will go for the patent. That should definitely go, because that's a novel process which you have developed. Yes. And converting that into nano itself could be another process which you can take up for the patenting. Based on these two products, now you can develop a strategy how to take up this into a kind of product formulation. It's not that you can directly take this nanoparticles without any formulation into the field application. Yes. So formulation development could be next strategy which you can take up, and then testing those formulation at pod trials and then the field trials. And also as mentioned by Dr. Sharma, where we can simultaneously also start doing the toxicological evaluation. There are prescribed for, if we go to the CIBRC, they have prescribed toxicological evaluation parameters. We can simultaneously go for the proposal for doing the toxicological evaluation. They can go in the this BIRAC as a proposal in the BIRAC, because BIRAC supports such type of startups, and they give the funding for this type of studies, even including the toxicological evaluation, the preliminary evaluation of the products. So once this stage is clear, then the next stage is obviously that's the entire thing, domain of the silica will be with you. So congratulations and my good wishes.
Thank you. Thank you.
Thank you so much, ma'am, for your nice suggestion. I'd like to just add that the field trials is the last objective of my SCRV funded project.
Yes.
So I'm going to just start the field trial on our agriculture farmhouse. So it is the last objective. So it is already in my
so
mind.
Sangeeta, can we have 30 seconds from you? You are the director of Amity Institute of Organic Agriculture.
She was named by Durgesh.
Thank you so much, sir.
Just press that.
You can, yeah.
So Sangeeta, you should give all facilitation.
Thank you so much. Thank you so much, Founder President, sir, for becoming part of this event. So Dr. Durgesh is very hardworking, and I have been observing him since last 6 years, and he has enhanced the graph of research of our institute. So I'm very proud of him. And other than this, Dr. Nutan Kaushik ma'am is there to guide him, and me also. And just I was thinking, how can I collaborate with him? Because I am a microbiologist and we have isolated some silica dissolving bacteria. So I was thinking, how can I collaborate with Dr. Durgesh and become part of his research as well?
Great. So now let us conclude with this note.
Okay, just a minute.
Yes,
संगीता जी, नूतन कौशिक बहुत तारीफ करती है आपकी.
Thank you so much, sir.
And to all present one here, I must tell you, once Nutan Kaushik showed the interest to join MIT from Terry, after long years at Terry, I didn't give any tawajjuh to that. Why should a person from Terry come to MIT? And 6 months, I didn't meet her. I didn't go to her office because I think she may go back. But now she's many, many years and she's a terrific scientist. And I call her Miss Multilateral. She has hundreds of contacts all over the world, Miss Multilateral. And she has now some very important projects with farmers all over the country. So I think we are proud to have such scientists here. So Sangeeta, you work more together, you and Dr. Kaushik. So I think you both and now Rupesh can do a lot of work together. Thank you.
Thank you.
Thank you so much,
Thank you,
sir.
Now the actions will be, first is field trials.
Yes, sir.
The second is establishing the safety and toxicity aspects, if any.
Yes, sir.
The next part is to file the registration for startup of this company, which AFSTIA will help. Dr. Neeraj Sharma and Dr. Chakravarti will help us. And these are all parallel action. So I would request a speed so that we have to give this product Atmanirbhar Bharat at the earliest possible. Let's congratulate once again.
Thank you so much, sir, for giving me wonderful.
Thank you very much.
Thank you so much, sir.
Move on. Yeah.
He will
You have filed the patent?
No, sir, I have not yet filed the patent because my field trial is still pending.
No, you can do it even at this stage. Wait,
Yeah.
Don't wait for field trials to come.
Patents are not for the field trial. Patents are for your process.
Yeah.
Yeah.
That process must be patented.
Okay, so
This is an immediate action. Within 72 hours, you need to do it. Goodwill, please note down this for action.
So I will monitor these activities.
Yeah,
So
please
I'll
ensure.
assure you, sir.
But sometimes the field conditions are different from the lab conditions. So I think your this thing has to be validated at the field level as well, right? So we can discuss and talk. Being an agriculture scientist myself,
yes,
we can talk about it and discuss and see how this idea can be taken up further.
Yeah,
right.
So the
Sir.
startup.
Sir, I have initiated all the work on the controlled conditions. So to whether know what is the impact of the at the seedling stage, and a lot of work I have done also on the agriculture field conditions. But the project as present is still ongoing. I have not yet tried the biogenic silica, and I'm very much sure that if I have found the results, my previous results, means silica nanoparticles and silicon is very effective on the field stage, then why it will not be a very helpful for the means biogenic silica will be not helpful at the field stage. So I'm very much sure that it will be very beneficial because my previous results, they have already, because it is only a modified form of the silicon, which I have already tested on the agriculture field trial.
Yeah,
so
thank you.
Sure, thank you so much.
Dr. Silvamurthy, sir.
Yes.
Sir, with your permission, a small clarification. Dr. Durgesh, I congratulate. It was not path-breaking. It was a revolutionizing presentation and work by you. Yes, because in your presentation, you had been using different species, right? From Pisum sativum to Oryza, to this khira and even the wheat.
Thank you so much, sir.
Does all these crops handle silicon in the same way or they behave differently?
No, sir. They have a same silicon. Only the concentration may be vary. Like in some plants like rice and wheat, we have applied 10 micromole silicon. It is very effective. But in maize and pisum, the concentration was 20 micromole. So it is a very little difference between only for the concentration. But the overall behavior, overall impact was very similar.
Okay.
Thank you so much, sir.
Thank you.
Thank
you
so much, sir.
Let's move on to the next presentation. Thank you, Durgesh.
Thank you so much, sir.
We invite Dr. Malika Chatterjee.
Founder President, sir. Thank you so much, sir.
Thank you. Assistant Professor, Amity Institute of Neuropsychology and Neurosciences, Amity University of Uttar Pradesh. And she's an outstanding neurobiologist, which Amity is very proud of. And she is a Wellcome Trust DBT India Alliance Early Career Fellow in 2013. And she is a PhD in Biomedical Sciences from University of Connecticut Health Center in US. And she is also Master's in Master of Science from Presidency College, Calcutta University. And she is a very, very renowned neuroscientist, which Amity is very proud of. And now she has been selected for Fulbright Scholar. Let's give a big clap to Dr. Malika Chatterjee, that she is going in the month of October to US for a very renowned institution. And Fulbright Scholar, to become a Fulbright Scholar is very rare. So we are very glad that she has achieved this recognition. She also brought IBRO grant to Amity University for organizing the workshops. And she has also been to Young Scientist Award she has received from the Society of Neurochemistry. And also the Singapore Neuroscience Association has honored her. So at this young age, she has achieved so much. Wish you all the best, Dr. Malika, to achieve many, many more accomplishments in your life, in your career. You should make some path-breaking research which you are going to present. Take it forward. Over to you.
Silvamurthy.
Yes, sir.
On Brain Talk, when Satish Reddy ji came, he showed a very great interest in her work. I saw that he stood and understood everything what Malika is doing. So Satish Reddy for DRDO is a great scientist, as you know. But when we were visiting and inaugurating the center, he showed a lot of interest in her work.
So Malika, these blessings are with you. Over to you. Go ahead on your presentation.
Thank you, Honorable Founder President and Silvamurthy sir, and hello to all the dignitaries. I'm going to talk about neurobehavior and using various model organisms. And I will just share a snippet of how we have been trying to use zebrafish as a model organism and how the story all started with me and my journey in neuroscience. So no coffee, no ideas. When I do an acknowledgement, I first acknowledge coffee. So without it, I wouldn't have done my PhD and my postdoc and even my, assistant professorship so far in Amity. So if you don't find me in the lab, you will find me in H block. And so coffee is the first acknowledgement. And then the acknowledgement slide goes to all the eminent scientists and the elite institutes which have helped me to grow into a neuroscientist in the past 12 years. So first comes my BSc college, where I did in graduation in physiology from Presidency University, Kolkata. Science College Masters in Genetics from Calcutta University. I went for a neurodevelopmental biology PhD, working on mice and chicks in University of Connecticut Health Center. My postdoc as a Wellcome Trust Early Career Fellow, again working on mice, was from the Tata Institute of Fundamental Research, Mumbai. And finally, not the least, I came to Amity University, Uttar Pradesh, Noida, and joined here as an assistant professor in 2018. So that was my journey so far, and I'm deeply indebted to any person who was part of this institute in my department and has helped me to grow as a scientist. So I will start a little bit of the story, and my favorite molecule, which I'll focus on later, is the heparin sulfate proteoglycan molecules. I used to look into a very specific brain structure called the thalamus, which is considered as a relay station in the brain because it basically sends information from the peripheral parts of the nervous system up into the cortex. And without having the thalamus, you can develop symptoms like hallucinations, because you basically do not censor out any excess information, sensory information. So basically, it's a very important structure. What you see here is a histological sagittal section of the mouse brain. And in green are the cells, GFP, expressing green fluorescent protein, is exclusively expressed in the thalamus in this particular transgenic mice that we created in the laboratory. It's a mice which is embryonic day 16 and a half, that is 16 days after conception. This is a coronal section where you see the same mice showing the thalamus and you see the thalamocortical projections going up into the cortex. That is the most important projections which carry sensory information from the thalamus to the neocortex. What we looked at was, since I was a neurodevelopmental biologist, I was looking at the development of the thalamus and the genetic networks that govern the development of the thalamus. So this is the mouse embryonic thalamus at embryonic day E12. And you see there's a small bunch of cells which express GFP. So how did we create this transgenic mice? We put GFP, this is a marker, which is under the GBX2 promoter. GBX2 is a transcription factor which is exclusively expressed in the thalamic domain and therefore only marks thalamic cells. We were looking into the GBX2 mutant mice at that time because we perceived that the loss of GBX2 might have an effect on the thalamocortical development. So what did we see? When we looked into the GBX2 mice, which showed us a gross defect of axon trajectory. That is, the thalamocortical axons, instead of going to the cortex, as you can see here on the right when we compare to the control, the green axons were going downwards towards the hypothalamus. Similarly, other defects were also found. That is, some of them went dorsally into the region called epithalamus, and some crisscrossed into another important region called the internal capsule. But none of these, or very few of these axons actually made it into the cortex. And if they did, like shown here, they went astray. That is, they went in various wrong directions into the cortical plate. So overall, in absence of GBX2, the axons travel more ventrally and are misguided in their positions in the cortex. This phenotype or this defect was very similar to another phenotype which was discovered earlier in 2007 by Lopez Bendito, who, using a technique called dye-eye, in which they put a dye into the thalamus to trace the thalamocortical axons, they showed that whereas in control from the thalamus, thalamocortical axons went to the neocortex. In a particular mutant, which I'll discuss later, which is ROBO1, ROBO2 double mutant, the thalamocortical axons projected ventrally instead of going to the cortex. This phenotype was very similar to the GBX2 mutant phenotype that we had seen in our study. And just to come to the explaining as to what ROBO1, ROBO2 is, ROBO1 and ROBO2 are in fact proteins. They are receptors which are expressed on the surface of thalamic neurons, and they bind to certain ligands called slit ligands, and which help them in deciding whether they will go to the cortex or they will travel towards the hypothalamus. When ROBO1, ROBO2 is there, the slit ligand will bind to that, and therefore the axons will normally go to the cortex as shown in this figure in the control. However, in the GBX2 mutant, we see, like the ROBO1, ROBO2 mutant, extensive projection of axons towards the hypothalamic domain. So they had a similar phenotype. So the question was, if GBX2 is a transcription factor, it regulates the transcription of various genes. So does GBX2 regulate the expression of ROBO1, ROBO2? And when you take away GBX2, you take away ROBO1, ROBO2, subsequently transcription doesn't happen. And therefore, the phenotype of GBX2 is similar to ROBO1, ROBO2 mutant. In order to understand that, we did an expression study using RNA in situ hybridization on sections of the mouse thalamus, where we saw in the control, this is the ROBO2 normal expression, whereas in the mutant, ROBO2 was downregulated. In the control, ROBO1 expression again was partially expressed, not into the marginal domain of the thalamus. However, in the GBX2 mutant domain, there was a huge increase of ROBO1 expression. This was unexpected because we had imagined in the GBX2 mutant, we would see a downregulation of both ROBO1 and ROBO2. However, in this case, we saw that ROBO1 expression was upregulated. This I have already explained earlier, that ROBO1 is a receptor on the neurons of the thalamocortical neurons, which binds to the slit homodimer. Now, what we saw, and we have already come to that conclusion, that GBX2 regulates the expression of ROBO1, ROBO2 differently in the developing thalamus. Subsequently, my PhD thesis culminated in the publication of 4 papers considering the genetic, revolving around the genetic networks of GBX2 function in the thalamus. And we showed that acting via transcription factor LHX9, GBX2 in fact regulates ROBO1 expression, and acting via LHX2 LMO3 loop, it regulates ROBO2 expression positively. But what we couldn't understand was if GBX2, in the GBX2 mutant, ROBO2 was downregulated, but ROBO1 was abundant. And as is known in other systems, ROBO1 can adequately compensate for ROBO2 function. So why wasn't it not doing that in the thalamocortical axon case and the GBX2 mutant? And why were we still seeing a mutant phenotype? So basically the question was, what compromises ROBO1 function in the GBX2 mutant, in spite of it being highly expressed expressed at higher levels than normal in the same? We brought in our thought of a third-party regulator of the action of SLIT and ROBO. So we hypothesized our HSPGs. What are HSPGs? They are heparin sulfate proteoglycans. Are HSPGs involved in regulating thalamocortical axon guidance? In order to understand that, we need to just briefly understand what HSPGs are. HSPGs are a combination of proteins and carbohydrates. Mostly they are expressed in the surface of the cell. They might also be free floating. Typically you get a protein molecule, which is found in the membrane, attached to the membrane, transmembrane protein. And to it are attached various carbohydrate molecules or carbohydrate chains, which consist of alternating domains of N-acetylated and sulfated domain. So sulfation is very important for HSPG formation. And this sulfation, as you see in the dots shown here, happens at various positions of the HSPG, which includes 2-orthosulfation, 6-orthosulfation, and sometimes, but rarely, 3-orthosulfation also. Now, all this sulfation, which is a very important part of the modification status of these HSPGs, happens through a specific biochemical pathway, which also involves, finally, the action of orthosulfotransferase enzymes. So basically, you need the expression of orthosulfotransferase enzymes in order for adequate sulfation to happen in these molecules. And why are HSPGs interesting? Because HSPGs do a lot of work within the cell. They can bind to the receptors and they can potentiate the aggregation of ligand molecules on the surface of the receptor and therefore enhance receptor-ligand interaction. So lately, just not receptor ligand, but HSPGs are also known to function in addition to receptor ligands in order to enhance any sort of receptor-ligand interaction. And we hypothesized, in our case, ROBO and SLIT might have been action potentiated by HSPGs. So what we saw was, in our GBX2 mutant, using microarray data of E12.5 embryonic thalamus, that all the enzymes which cause HS6ST sulfation at the orthosulfate group are significantly downregulated in RNA expression in the thalamus of the GBX2 mutant. And this we also validated in our sections, and this is a manuscript which is on the preparation. We saw HS6ST 1, 2, and 3 expression downregulated in the thalamus of the developing mutant. Now we looked at the HS6ST mutants, the mice mutants, to see what is happening. Is it something like the GBX2 mutants also? And here you see ST1, ST2 double knockout, in which what is happening is we saw that there is a complete absence of thalamocortical axons in the ST1, ST2 double knockout, suggesting that from the thalamus, cortical axons are not reaching the neocortex. We did a culture experiment, explant culture experiment in vitro, where we took thalamic explants. They all have small neurites here, as you can see. And these green mass of cells are COS cells, which express GFP in this case. They express the ligand for ROBO, that is SLIT1, in this second experiment. And this is a double knockout, that is the heparin enzyme sulfate knockout experiment, where again the COS cells are expressing GFP. And in this case, the GFP cells are expressing SLIT1, and the explant culture is from a double knockout mice. You just compare this data, and you'll see, whereas in the presence of SLIT, the wild type neurons from thalamic neurons just totally repel, which is their natural activity. They repel from the source of SLIT1 and move towards the other side. Whereas in the condition where the orthosulfate enzymes have been knocked out, the slit ones, the thalamic axons don't care. They move equally away and towards from the slit source. So that was that this, in absence of any sulfation enzymes, the thalamic axons do not know where to go. That was the conclusion. So HS6ST1 and 2 are essential for the responsiveness of the thalamic axons to the slit ligand. We did further in silico studies when I joined Amity with my colleagues, Drs. Vijay Kumar and Amrish Kumar. And we wanted to understand what is the interaction of slit, robo, and the HSPG on the thalamic neuron cells. And since we couldn't do such an in vivo study, we went for an in vitro study. And all the 4 energy subtypes which are involved in the protein-protein interaction, we saw was significantly downregulated in value in the mutant, suggesting that there was a loss in energy in the interaction of the components in the mutant complex, indicating a functional disruption in the function of slit, robo, and HSPG. Further, they adopt, going by our root mean square deviation analysis in 500 nanoseconds, and also the RG analysis. What we saw, that what you see in red is that action of the mutant or behavior pattern of the mutant complex. And what we concluded that it adopts a less stable, the mutant complex, that is orthosulfation lacking enzymes when present, adopts a less stable and more flexible conformation of slit and robo. And finally, by our PC2 analysis, we also saw that the mutant complex covers a larger area of conformational space than in the control. So our conclusion based on the in silico analysis was the lack of 6-orthosulfation in heparin sulfate moiety causes loss of stability, increased flexibility, and subsequently the loss of function of the triad of slit, robo, and HSPG. This was a summary. And loss of ST1, ST2, that is the sulfation enzymes loss, partially phenocopies axonal guidance defects of GBX2 mutants. A subpopulation of the thalamocortical axons are directed towards the hypothalamus. Further, we have seen, but have not shown here, that axons are reduced in number towards the cortex, and axon bundles from the prethalamus, which is a region ventral to the thalamus, are shifted laterally. So this is the model which we have. GBX2 as a transcription factor, inhibits ROBO1 expression, upregulates ROBO2 expression, controls HS6ST1, 2, 3 expression, and together...
Malika,
yes.
Malika, can you also relate to the application, this knowledge
Yes.
which you have generated
Absolutely.
in the health and disease? How
Yes,
you
I
will
will come
use
to
this
that.
model?
Yes, yes, certainly. So these will together, so the RoboSlit HSPG is just a model system to show how HSPGs can be involved in RoboSlit interaction. But to remember, HSPGs are not only involved in RoboSlit interaction, they are involved in other ligand receptor interactions also. And as Sir said, I'll just quickly show this. It has been known that heparin sulfate loss or lack of sulfation of heparin sulfate molecules within the brain can lead to various types of disorders, including autism and schizophrenia. Okay, so there are many published records, and we are therefore interested into looking what is the role, as a fundamental biologist, what is the role of heparin sulfate proteoglycans in mechanisms, neural mechanisms in cell formation, cell development, and neurobehavior subsequently. We are also interested into looking at what mimetics of HSPGs can be used to modulate HSPG behavior, HSPG action within the cells in order to make the neurons, like in RGBX2 mutant, for example, better guided towards the cortex. So these are very fundamental questions which we are still addressing. We are at a very fundamental stage, but that is also the reason why I wanted to introduce HSPG, because with the guidance of Professor B.N. Malik, we have just started the zebrafish lab in a year. And one of my goals is to look at HSPG role and the sulfation roles using zebrafish as a model organism, because it will be very easy to track neurobehavior using the zebrafish. And that just gets me back to this slide, using other model organisms to study the role of HSPGs. Because I was talking about sulfation so far, right? So there is a concept called heparonome, which I have not put the term here. Heparonome means the multiplex population of heparin sulfate proteoglycans that exist in every cell in the human body. And it is known that a varied amount of sulfation of heparin sulfate proteoglycans can contribute to an altered gene expression pattern and subsequently different behavior of the cell types, depending on where they are expressed. So it's like the heparin code. If you change the sulfation pattern of the existing HSPGs, you can change the function and the activity of those particular cell types. And there are a couple of papers which have published, shown that. So, because zebrafish can be used to study dose-dependent studies a lot, so and in a large throughput system, this is one of my model organisms of choice as compared to mice. When we start to look at small molecule mimetics, as Dr. Silvamurthy mentioned, and see what is the effect of changing sulfation in the brain of using the fish as a model system. And that brings me to my Fulbright objective, which was just formulated 6 months back. And as you can see, the objectives we have just started. We just got the grant and I have just started it, but I wanted to share my plan so that I will also have some roads and insights from the experts here. The objective first would be to analyze the extent and pattern of CNS, that is central nervous system, HSPG sulfation in developing and adult zebrafishes exposed to small molecule inhibitors of endogenous sulfating and desulfating enzymes. And that is what I think can have direct translational benefits, because I have selected a couple of molecules, obviously not a very large cohort, but the best known molecules which can be used to change sulfation in the brain, simply put. The second objective, which I will be primarily doing in the US, in Professor Robert Brown's lab, Professor Robert Creechan's lab, is to determine the effect of reduced or enhanced HSPG sulfation on neurological behavior in the developing and adult zebrafishes. So basically, if we find a difference in the neurobehavior on changing sulfation patterns, then we'll go back and work on those molecules and see what cellular mechanisms can explain the change in the neurobehavior. But first, we need to see at a broad scale what effect these small molecule inhibitors have in neurobehavior. And finally, as a broader goal, and which is also as the Fulbright application mentioned correctly, is what are you bringing back to India? So as part of my goal and a bigger objective and vision, I want to establish a high throughput larval zebrafish larvae neurobehavior imaging facility at AINN in Amity University, Noida, for analyzing larval fish behavior when exposed to various small molecules, morpholinos, etc. And again said that broader goal, it is not limited to my HSPG molecule inhibitors alone. We also want to look at other molecules or other questions that we have and see how behavior changes using large throughput formats. And this is something which my fellowship, thanks to the Fulbright organization, Fulbright Commission in India and USIEF and the Government of India and Amity University, will allow me to travel to Professor Robert Creechan's lab in Brown University. So what has he done? He has actually a very revolutionary scientist in the field of larval imaging, where he is preparing cost-effective methods of imaging a huge number of larvae, like 384 larvae right now, at one go with a simple DSLR camera, and to acquire the live feed and then process it using freely available softwares to analyze various movements. Like, for example, if you give them a sound stimuli and see how they respond to the sound, because these during early development in various disorders like autism and etc., early marker of movement and all become very important to track. And this tracking will also become a part of the application that can be extended to mice and humans eventually. Like he has published this analysis of vertebrate vision in a 384 well imaging system. And this is the setup that he uses using a camera. And this is the simple 96 well plates. And you have total 384 larvae in them. He uses an LED projector and various acoustic and visual stimuli to track as small organisms as this. This is the example of a fish larvae. Now, in continuance of that, like what were we doing so far? Well, I was working on mice, right? So we had to do something, set up the zebrafish lab to address this and other questions. So that brings me to what I've been doing since the last 1 year. We have been trying with Professor Malik's guidance and obviously generous Amity and IBRO, International Brain Research Organization funding. to set up the zebrafish neurobehavior as well as the histology lab at AINN. So I'll just show you a couple of, I'm hoping this video will work. We are tracking, this is not working. This. I thought this was said, sorry. This was a video thing. We are tracking adult movement and let's go on. Next, okay. Okay, I got it. Next. Okay. So this is using Animaze software, which we have, and this is the actual fish in a tank, in a glass jar, and this is the roadmap of where it is going, being tracked with an Animaze software. This is a apparatus called the T-Maze, which is used to track behavior by training the fish, after training the fishes. I'll come to that in a bit. We have since I'm a developmental biologist, we have been trying to grow the fishes, so these are the eggs. And this is a 2-day-old larvae, and this is an alive 4-day-old larvae. And so we have, since we want to follow from development to adult, so we do all stages. And we track, we are attempting to track at all stages as well. Just coming to a little bit of the data, preliminary data that we have generated as a part of the neurobehavior experiments. As I mentioned, this is the T-Maze. We have developed this T-Maze facility where you have this instrument in which that it's filled with water. And this is the start zone where you put the fish and then you let it roam around freely. This is the green zone and this is the red zone. This is marked by us. And green zone is a reward chamber, red zone is the punishment chamber, wherein how does the fish know this is a reward chamber? The moment when we train, during the training part, when the fish goes into the green zone, we give it food. When it goes into the red zone, we give it a mild shock. So by a couple of trials of training, the fish knows where to go. A normal fish, a trained fish will only go mostly into the green zone and not into the red zone. And this is the typical heat map generated by Animaze to show the movement of the fishes and where it's staying for most of the time. This is a summary of 24 hours of data. This is the training phase, a green zone and red zone, and you give electric shock. So this is part of the passive avoidance paradigm to train and test fishes for spatial memory using the T-Maze. And a very, enterprising intern who came to work with Sir and me, Ishitpreet Singh from Aisen Mohale, actually came up with a brilliant paradigm where he combined this T-Maze protocol with a sleep deprivation paradigm. The sleep deprivation protocol has been designed in Professor Malik's lab and with a very energetic student called Kumar Abhishek from JNU, who has actually taught us how to do this, So this is the sleep deprivation chamber where there is a CCTV camera and you put the fish in a jar here. There are you can put in 2 jars at a time and you have bright white light. Okay, and greater than 150 lux. And if you train when you keep these fishes in the jar, you expose them to greater than 24 hours of light, the fish develops is highly sleep deprived. And due to constant light, they do not sleep. So you have a constant sleep deprivation protocol achieved. So what Ishitpreet did was, in day 1 in T-Maze, he habituated, he let the fish move around in the T-Maze, know what the T-Maze is. In day 2, he did training and recorded the fishes. In day 3, he sleep deprived the fishes, but for different amounts of time period, that is control or 24-hour total light, that is chronic sleep deprivation or fragmented sleep deprivation. And what he saw was, I'll quickly come to the conclusion, was as in the control, in the trained fish, when there was no sleep deprivation, went more to the green zone, which was the reward zone. So it remembered that it had been punished in the red zone. However, when it when it was sleep deprived or fragmented sleep deprived, it forgot that. And therefore, there was no significant difference between the red and the green zones, whether the time period of sleep deprivation varied, but they still didn't go. The speed, however, wasn't significantly affected. Again, we did a modification of this experiment, which is still ongoing with our talented BSc student, Meet Seth, in the BSc 6th semester. We are trying an enriched environment. Enriched environment always is known to potentiate memory. Okay, so whether the training in the enriched environment actually is potentiating the fish to remember more to go into the green and avoid the red. And this is something which seems to have an effect because as compared to the control group without enriched environment and with enriched environment, there seems to be a more significant choice of the fish to go towards the green environment. And there was, as expected, no significant difference in the sleep-deprived group. But again, this is very preliminary data. Neurodevelopmental disorders, because I work on neurodevelopmental disorders, we are going to look at larvae. So these are tracking of larvae as less as 4 DPF, that is day post-fertilization. We can start tracking with NMAs as less as 4 DPF. And this data that you see is actually from a 7 DPF fish. And this is a 96 well plate in which we have marked inner zone and the outer zone of the plate. Why did we mark the inner zone and the outer zone? Because the inner zone, if the fish stays for a long time in the inner zone, that is, it is comfortable going around exploring. But fishes who are anxious tend to stick to the outer wall of the plate. And therefore, that is a behavior which is called thigmotaxis. And I was setting up a model for autism because that is one of my key areas of interest. And we use various models, various concentrations of valproic acid, otherwise an anti-epileptic. It is known to cause autism spectrum disorder. And thigmotaxis behavior, like as I said, was checked at the larval stage. Basically, we checked whether these larvae would remain in the inner zone or remain in the outer zone. So we compared the two zones. And what we saw was at various concentrations, when we were testing, the time spent was significantly more in the outer zone in case of 25 micromolar and 48 micromolar of valproic acid. That showed, that means that the larvae with higher concentrations of valproic acid had higher signs of anxiety. Okay. We have set up another system of testing called novel tank test, where in this jar, we actually demarcate it into 2 areas and put the fish there. And then, sorry, this video is not working, sorry. We calculate whether the fish is going into the top portion or the bottom portion, remaining in the bottom portion. Remaining in the bottom portion means the fish is not exploratory, it is anxious. If it goes into the top part, it is exploratory. Okay. So what we saw was both the time spent in the upper zone as well as latency, that is whether it enters, how long it takes for it to enter in the upper zone, was significantly affected. Where at concentration, we did showing the data here for one concentration, that is 48 micromolar, that's the maximum concentration of valproic acid that was used by us. So, and this is for adult. The previous one, anxiety test, was used for the larvae. So we set up 2 test systems basically. And we also did the predator avoidance test, wherein you have a predator like a big fish in one part of the tank. You have various barriers here and you put the zebrafish at the other part of the tank. And even though we need to put the barrier, or else the predator fish will eat up the zebrafish. But we just check by seeing the predator fish, how does the small fish, that is the zebrafish, react. So there were 4 conditions: zebrafish with transparent barrier, predator's presence, zebrafish with transparent barrier without predator's presence, opaque barrier with predator's presence, opaque barrier without predator's presence. And each prey fish movement was again video captured with NMAs and tracked for 6 minutes each. And this is a representative software, the heat map, which shows that a typical fish on sighting the predator, which say is here, will tend to stay away from it. So that shows that there is fear in the fish. So this is a typical test, again set up with the help of Kumar Abhishek from JNU and with Sir's guidance. So very preliminary data, but I hope that we are able to now really start doing experiments, that the projects we have planned, and also my own heparin sulfate experiments, which I'm going to learn to do neurobehavior larval imaging in Professor Roberts' lab. And my broader goal actually is to set up the high throughput neurobehavior imaging of the larvae for analysis of various early sensory markers. That is, how do the fishes respond to acoustic stimuli, light stimuli, color stimuli, because in ASD and early neurodevelopmental disorders, tracking fish at or any animal at the early stage is very crucial, and there are no markers, validated biomarkers available in the field as of now. And if I narrow down to my own selfish goal, that is exploring the role of HSPGs in the brain development by selective manipulation of sulfation pattern of HSPGs in vivo. And this is the latest. This is a group picture we took from 102 Fulbright scholars, right from the masters to the faculty fellows like myself. And that was in Kolkata last week. And we are really proud to have Amity on the map. Thank you. Thank you to everybody. Thank you.
Malika is again a fantastic model development for studying the neurobehavior. Because normally you only look at mammals, the maze studies in animals. Now you have gone to zebrafish and also to C. elegans. Again, you are doing some work on C. elegans. And in the zebrafish, the larval behavior also you are looking at. These are all wonderful models which will be very beneficial to study the neurobehavior in health and disease. And also to try some drugs which can counter sleep deprivation effect on cognitive behavior, memory, concentration, learning efficiency, psychomotor performance. So many things can be studied using these models.
So, I would just like to mention, because this high throughput imaging is very useful for many types of disorders. Fantastic. And so that would actually, because the dose dependent studies and all could be really done very effectively. And as Professor Cretan has told me and promised me that I'm going to show you more of my techniques once you are here.
Yeah, learn in this Fulbright. It's a very great honor for us, Amity, that you are a Fulbright scholar from Amity. Now we will move on to our experts. Dr. Pankaj Seth, he is a professor, very distinguished professor in National Brain Research Center, NBRC. And he has particularly contributed in HIV dementia related to human immunodeficiency virus, HIV. How does it cause dementia, the cerebral effects of HIV. Then he has worked on Zika virus induced the microcephaly in infants by altering microRNA circuitry. That's another area. Then in COVID, many people, they suffered brain fogging. So what is the process underlying this neurophysiological or neurobiological basis of this? Then he is also fellow of the National Academy of Science, National Academy of Medical Science, Indian Academy of Neuroscience, and he is a member of large number of task forces in ICMR, DBT, and other funding agencies. And he is also co-chair for chronic disease biology and neurobiology of DBT-TEC. Now over to you, Professor Pankaj Seth. We want to hear from you these models which he has developed, and what are your guidance and advice to Dr. Malika, our young, brilliant faculty.
Sure. Thank you, Dr. Silvamurthy, for adding me into this very important event. I think all institutions should have these. First, to bring these bright young researchers on a platform where they can express their usefulness of their work. And I would congratulate. I've been listening to all the researchers so far, and I'm really proud that Indian science and the youngsters are doing the very good work. So for Malika's work, of course, this directly falls into our domain, and that National Brain Research Center, as you said, we are also interested to understand how healthy and disease brain work. The model system which she is working on is wonderful. Zebrafish has a lot of advantages, and I think the high throughput screening for identifying newer molecules is the way to go. So I think she's on the right path, and I'm aware of her work. I followed her work. I think I've interacted with her also in some of the meetings. She will have to remind me where I met her, but she's a very bright student, researcher. Now, a few point of advice to Malika. We are now in a stage where we can advise as well, and so I would suggest that Malika, if you could think of when you're training your fishes, right? So, we know that memory consolidation occurs during sleep. So sleep deprivation definitely is going to affect it. So, but your data is also showing. So have you tried during their training also to introduce sleep deprivation?
Sorry, can you just repeat the last part once?
So have you tried adding sleep deprivation during their training?
Oh, interesting.
Okay, that's one thing which you can think of. Also, definitely autism is the way to go for this because it's a hard wiring defect, which occurs during development. So I'm not surprised that the GBX, mutants are linked with the absence of GBX or changes in ROBO1 and 2 are linked with autism. So what are your, how do you think you're going to apply this into understanding autism? Because ASD, I would just like to tell you that there's a multicentric consortia being developed between AIMS, NBRC, NHANS, and a couple of other institutions. So I will be happy to, by the time you come back, I think it will be in place. And then you can get in touch with us and we can also rope you into that because we do need some basic aspects of it. I think you can feed in there. While we have clinicians like Shifari Gulati and others who would be doing the main job.
Thank you, sir.
And at some point, you should also think of validating all this in human tissues. Zebrafish is a wonderful model. It gives you an advantage of all those experiments which you are planning. They're not possible in humans. They're only possible with in the model system you're doing. But if we are looking at the gene level and all that, you have to validate. So the way we had done also was we started with human cells, and wherever possible, you should get tissues from the brain bank. I'm sure you know about the NIMHANS brain bank.
Yes.
Dr. Shankar had started. Now Dr. Anita Mahadevan is taking it forward. When you're going to US, you have an opportunity to look at or get in touch with people like Ted Abel and others at Iowa Brain Bank. And, have these contacts, develop these contacts. They're going to help you. Networking is another key, which you could see from your predecessor, Durgesh's work. See how well connected he has placed himself so well. He's connected that way. So this is what my piece of advice to you, and you're doing wonderful work. And Dr. Silvamurthy, I think MIT is on the right track on this part. And of course, with the addition of Professor Malik, I think, the neurobiology and neurosciences group is also going to flourish there. Thank you so much.
Thank you, sir.
Thank you, Dr. Pankaj. Yeah, please.
Curious to know, can we use this as a marker to detect the autism? And is there any way that we can reverse this effect?
Oh, I'm sorry. Which as a marker? HSPG?
The HSPG.
I wouldn't say at this point. We still don't know. Okay. Yeah. And I didn't get the second question. Can you just
can we reverse this effect by any means? What you call
autism.
No.
Again,
the heparin
so that is one of my things that I want to first find out at the fundamental level, how the sulfation changes with the small molecules is causing neurodevelopmental behavioral changes. And if we can reverse by increasing or decreasing, it's a full study right now.
Okay.
Yeah.
That's great.
Yeah.
Thank you. Now we move on to the next expert. I think you should connect Malika with Dr. Pankaj said.
Yes, I will.
That is the premier institution for the basic and applied research in neuroscience. And please, NBRC is a very reputed institution, and please connect with him.
Certainly, I'll do.
Now we move on to the next expert, Dr. Usha Panjwani. She is from Defense Institute of Physiology and Allied Sciences. She was heading the neurophysiology department there. And she has worked on sleep deprivation and cognitive performance in soldiers who are during war. They will be sleep deprived, totally sleep deprived or partially sleep deprived. So how it affects cognitive functions. She has used Mori's water maze. She has studied the role of hippocampus. She has also done some behavioral testing in animals. So over to you, Usha. What are your thoughts on this presentation as well as for Malika? Over to you.
Thank you so much, sir, for this opportunity. And my greetings to all the, the fraternity of MIT as well as the founder member. I must congratulate Malika for this very nice piece of work which she's done. And basically what I had some questions in mind, but over her period of presentation, she's answered all the questions. Very good. I'm so happy that you're doing so well, and Amity is doing so well, and over this achievement of Fulbright also. And very interesting that you have chosen fish as a model for this study on sleep deprivation. So basically, just a small query. How did you confirm that the fish was actually sleep deprived for that particular duration which you are targeting?
Yes.
Have you checked it?
Yeah, that's a good point. We have taken the fish and we have just, we'll do cortisol and levels analysis on that. We haven't yet done that. Yeah.
Done the polysomnography study?
It would be a test for the level of, or noradrenaline also levels as per Sir's publication, Malik Sir's publication. We can check their levels. It's supposed to go up during REM sleep. He might be better to comment on that. But yeah, NA, noradrenaline and cortisol levels would be two things we will look at if we want to validate.
So why is it a preferred model, fish?
Oh, fish. It was my preferred model, then it was Sir's preferred model because first cost effectiveness and also drug use, it is so easy to do, again, high throughput sequencing. Eventually, for example, I'm not high throughput sequencing, high throughput neuroimaging. Cost effectiveness, the most important thing. When we talk about, working on so many mice and giving them these drugs, at the first go, I don't think at the current status, how things are, it is too ambitious or not worthwhile to directly start with mice, especially when a person like me, wants to plan a lab. And fish, as is already been shown by Malik Sir and other fish studies on sleep deprivation, it's a very good model to show as a sleep deprivation model. So,
Thank you.
Yeah.
Thank you, Dr. Usha.
All the best to Malika. And
thank you so much.
We also have two industrial experts who are going to collaborate with Dr. Malika. So what is that, Malika, you are looking for? Then I will first listen to Dr. Vigyan Mishra. He is chief of the lab, northern part. There's a Newburgh Diagnostics. So he is there as the chief of the lab. I like to listen from him. He is a MBBS gold medalist and MD pathology, doctor of medicine in 2021. He did his dissertation on the expression of cyclin D1 and P53 in oral squamous cell carcinoma, and he also a fellow of the clinical cardiology, and he has also been in Apollo Hospital's Education and Research Foundation. They have recognized him. And so he's a brilliant person. And now I like to listen from him that what is his association, which Malika is planning with you. And then I will also listen to the other expert also. Over to you.
So, first. Yeah, now it's okay. So, firstly, I highly appreciate the model you have chosen and the work you are doing, and it has great importance in medical science per se. I will say the domain I help with. So, the vision that you have is very clear, and the model you have chosen is very workable. The thing that in medical science, when we are working on a model, we see the final outcome or the final output per se to diagnostics, like the biomarker, how it will be helpful in preventive science for early diagnosing a disease, or secondly, when there are clinical presentation in the patient, at that time, how we can find out or how we can establish that this is the clinical scenario. And thirdly, when the patient is on an intervention, like on a medication, we also monitor the complete disease progression or regression, whatever it is. It is concurrent to the question indirectly Dr. Nutan asked to you. So definitely you are understanding the behavior in the model, but I hope you will take it to humans also. And when you study, definitely it's a challenge to study in humans. So I would suggest there are organ on chips model to study the drug intervention. So concurrently, you may also adopt one of them.
Right.
And establishing the genetic biomarker has been suggested by an expert is extraordinary tool in which you can take it very early in the medical scenario. But I'm very like positive with the work what you are doing, and definitely I see a good collab and very good biomarker coming in the market, because autism or schizophrenia, these are very complex disorders to even understand, diagnose, and we have around 2,500 neurologists working in India. They cater more than 10 million active cases, and definitely newer deliveries. We need to screen with ultrasound imaging. We are screening babies. We are concurrently doing genetic study for autism. We are establishing biomarkers. So in similar fashion, I would suggest you to establish a correlation, and let's go with the path breaking discovery that you are making.
Right. Thank you for your advice, and we'll be in touch. Yeah.
Yeah, sure.
Thank you.
Thank you. We also have another expert, Dr. Vishal Nanavati. He is a DBT Ramalingaswamy Fellow and consultant senior scientist of genomics medicines at the Newburgh. And he has done his PhD in regulatory biology with specialization in cellular and molecular medicine from Cleveland State University, Ohio, USA. And he has got several skills, professional skills over a period of time. So I like to listen from him. What are your thoughts on this presentation, on the work, and how you would like to, Newburgh would like to associate with this study?
First of all, I would like to thank founding president and management here to invite us for this particular seminar. The good work, basically, there are tons of questions that I have at the basic science level. Will fragment it over the two different segments. And first of all, I also would like to congratulate Dr. Tripathi and Dr. Malika, right? Sorry, Mulisa, ma'am. Yeah, I can see like even a cross combination just within this room right now, right? Like the click chemistry can be working for the plant segment, and the silicosis can be working for the neuroscience as well, right? So, yes, there are questions like how to go and address about the basic neural changes in general, and what Dr. Usha asked, I also had the same question, like the sleep deprivation. How do you know they lost their energy to travel up, right? And whether you would question that or not, and whether you're going to change it or not. Regarding the zebrafish, I had an opportunity to work with another scientist in Cleveland Clinic, and I've helped them at their high throughput sequencing scenarios. So yes, those high throughput images along with the transcriptomic profiling, proteomic profiling does gives us answer. And yes, with the help of Newburgh and the facility that we can have in execute at the deliverables that we are looking for, right? Like I think we are very confident that we can help you out, and rather you help us learn more science as well, because I think we are really looking for industry academia collaborations in general. In terms of couple of suggestions that I have, and pardon me, I'm very naive in neuroscience in general, but there is gut-brain axis that has been spoken widely about in autism. And autism is not just one. Now we are saying as an autism spectrum disorders, right? Somewhere do you think we can fit that with zebrafish model as well?
That's an interesting thing, because my collaborator, Dr. Devinder Toor, he's an Amity faculty. We have just initiated a project where in sleep deprivation conditions, that was one project, we are looking at the gut microbiota changes in the sleep deprived zebrafishes and comparing them to the control, because it is all known in sleep deprivation, gut microbiota is affected. Okay, and what are the changes in various conditions of the various types of sleep deprivation is what we are seeing. And since I have already the valproic acid model, chemically induced autism, that would be my second proposal. And in fact, we just put in an IAC proposal for ASD model gut microbiota analysis with, again,
I think Dr. Hridesh Prakash would like to supply.
So I think you have raised a very fundamental issue, gut-brain axis, and because it is heparan sulfate, glycoprotein, heparan sulfate is a ligand for the TLR2 and 4, which this we actually take it from the cancer immunotherapy point of view. So coming back to the gut brain as the third brain of the brain which is now well accepted phenomena by the biologist community. So this heparan sulfate glycoprotein would be helping us in sensing the invading bad microbiome if in the autism condition and disease condition where the blood-brain barrier is leaky, for example, and this contribute to the infiltration of the immune cell. And that is where I think you can link up with the immune intervention.
Yeah.
And I think Devendra Toor can probably help together with us.
We are
targeting the microglia. We are working on the macrophages. So by targeting the infiltrating immune cells, you can very much control and tune the sleep phenomena. This is my proposal from immunological or translational immunological point of view.
Yeah, so that's fantastic, right? Because ultimately the zebrafish model needs to correlate back for the evolution of the human brain and the disease models. So yes, this cross collaboration helps. Another thing I think I would like to add to this is that do not hesitate of thinking even larger than this, because now single cell technologies are available with us. Which is basically single cell genomics and spatial transcriptomics. We can perform it. It is right there in our facilities, and we've been doing it now. So we can definitely help you out to figure further things around this. Yes.
And moreover, we are working on the animal model, the mice model. So that will add up to the whole program which Malika has initiated.
Yeah, but overall, I think we are looking forward to hear more from you, and we will be happy to collaborate. And
thank you so much. Yeah. Same.
Thank you, Dr. Vishal. Thanks, Dr. Gopal, you for bringing these two experts from Newburgh. Now, I'd like to listen from Dr. Malik, B.N. Malik, the head of the director of that Amity Institute of Neuropsychology and Neuroscience. And she works with him. So he is an expert. He is a Bhatnagar Awardee. Dr. B.N. Malik is a Bhatnagar Awardee. There are only 6 experts globally known for REM sleep. So he is one among them, and he is the only one living. All others have passed away. But we are fortunate to have that person who has contributed immensely in the field of REM sleep, sleep deprivation, and neurochemical basis of sleep deprivation and sleep, REM sleep. So all that area, locus coeruleus, all those areas he has worked. Over to you, Malik, that you have been part of this. You have helped her to grow to this level and groomed her, motivated her. We want to listen from you. How you would shape her further, and how do you translate this into application in autism or the epilepsy, or it is in the neurodegenerative disorders? How would you associate HSP?
Okay, thank you, Dr. Salavamurthy, and thank you all the speakers. See, the way I look at it here, my job here is to develop the department. And here, when I joined about a year and a half back, we have excellent, talented young faculty working on 2 reasonable neuroscience subjects. My job is to develop and help each of them. So once I thought that, first thing, biology is a derived subject, and for neuroscience, any part of the work in any biological subject will be applicable there. In addition, at least 3 more subjects are needed: anatomy, behavior, electrophysiology, and mathematical modeling. And fortunately, working under this condition, I think under one roof, I had developed each of these in 37, 38 years in JNU. So that helped me. And that is how I could develop here. One example is Dr. Malika. I am grateful that what she's saying that I have done, but it is all what she has done. I tried to do whatever my bit. Like that, I must take this forum and help. There are 8 more faculty. Each of them are developing in a nice, fantastic way, right from molecular modeling to neurochemistry to electrophysiology and all these. So therefore, I would like to whatever little bit I can do. And model of zebrafish, yes, what Malika said, in addition to that, not only cost effective, is very well controlled. We can do the genome is almost 90% conserved. Behavior can be done. The brain, each of these, what we get in the human, that has been represented. The anatomy precisely also has been done. And as regards what Usha, well, Dr. Usha, I hope Usha don't mind if I say Usha, okay? She was doing MSc when I was doing PhD there. So Usha, you may remember the stereotaxy, what I had to take your class. So there, electrophysiology, there are only one person. We are the second person who had done EEG in zebrafish. The student has yet to submit the thesis, maybe in 2-3 months, will be my last student at JNU. But yes, how do you do? Many people have talked about sleep deprivation in zebrafish. That is by recording their movement. So Malika, you have also done 24 hours recording of the movement. There we checked. Now there are more science to it. Now I think we are getting late and there's another speaker, we can talk there, but only one word one sentence I will add, possibly their unihemispheric sleep. Those who understand, one can go about, but otherwise, but we can check all those things. With this, I think I'm thankful to Malika, who has worked very hard to bring this to this level. And congratulations for your Fulbright Fellowship. And at the same time, I must thank the entire Amity organization, right from founder president to vice chancellor to Dr. Selvamurthy, Dr. Das, and Dr. Rutan Kaushik to support with the instrument. And then we are trying to develop. Thank you very much, everybody.
Thank you.
Thank you, Dr. Malik. And we have a lot of expectations, aspirations from Dr. B.N. Malik with a rich experience that he will develop this as one of the centers of excellence in neuroscience in our country. Now, the last presentation we want to postpone, because the reason is already it's late, and next time, I think goodwill, we should restrict to only 3 presentations or 2 presentations so that we can have a lot of discussions. And we can have more frequently. So maybe every month we can have one. So that we will have every 2 months, 4 people we would have covered. Then we can discuss it at length. So let's not have 4 presentations. Let us have 2. That is another. So I would request Dr. Professor Andrew Lin. We have requested him and he has been also been listening to all this. But I want to listen from you, not on this presentation which is going to be made. Dr. Ravidatta Sharma, we are postponing your presentation. We have spoken to you and Dr. Goodwill has spoken to you and you have kindly agreed. But I want the permission of Professor Andrew Lin. He is Professor, School of Computational and Integrative Science in GNU, and he is an outstanding scientist. So, Professor, we want to hear you. Even though we are not going to listen to the 4th presentation, we will request you again to come. But over to you. What are your impressions from JNU? You are an expert in many fields. So in the field of life sciences, over to you.
Professor Selvamurthy, can you interrupt before Andrew says?
Please, Rajendra Prasad. Yet another great scientist, Dr. Rajendra Prasad.
Andrew Lin is a very humble person. To catch him for this long was a big ordeal. And he has been sitting here patiently. So I don't know what will be the next time, whether we can get him for this long. So I know
if I can apologize for my lack of communication, I've been caught up with a medical issue in my family, but I cannot stop from attending something that's associated with Ravi's work. Dr. Selvamurthy, first I should express my admiration to the founders and those who created Amity University, because I watched it grow over the last 30 odd years. And it has served as a space for you. I can see Professor B.C. Das. But I would like to put on note, I mean, you mentioned me as someone from JNU. The people that I really look up to in JNU are now in Amity, Professor Upadhyay, Professor Ajit Verma, and on this panel, Professor Malik. And I would explicitly, with reference to Ravi, talk about Professor Rajendra Prasad, who's at the other campus. Now, see, the thing is that I have constantly worked with biological data. And now biology is data driven. And Ravi's work would have taken the three other really excellent talks and would have shown you how you could actually be powering data driven biology and the bioinformatics and the area that it is. So I will not, since it would have been nice if I could have, he could have introduced his work, but let me just very briefly talk about it since you've had these great mentors. Ravi, to me, when you talk about the 5 pillars that you were product to processes, etc., and path breaking, I think the path breakers and how your environment creates such path breakers is also something to explore when you're talking about institutional building. And Ravi was a young MSc about 15 years ago when he started teaching at the Meerut University before I linked him up to a colleague in Spain. And I have done only that. He had published papers as an assistant professor while teaching and has driven his work with omics about 12 years ago and is now leading a data science program at Amity University, which I have to say is among the best programs in the country. Because of the vision at which it was set up. No, it was because of the vision at which it was set up and the timing. Now, we're all talking about ChatGPT and generative AI. You are teaching it in that campus, in a biology department. Now, this is so rare worldwide because you have people who are 30 plus, who are experts in that area. So I think you should actually look at not just the resource-driven data that you're generating and the kind of work that the 3 excellent presentations have made, but how you're going to take this forward and how you're going to link this up. And that was where Ravi's kind of work and the department that he now, I think, is heading, I'm not sure, because one of the other colleagues has moved on. There will be a high attrition rate on data science. His work is essentially on using these kind of methods to detect alternative splicing, which is a very biological topic. But when you look at it mathematically and computationally, I think I was reviewing a thesis of his, so it was published in January in the highest journal in bioinformatics. Briefing is in bioinformatics. So I wish we could have listened to him, but I understand your time is of a concern. But I would like to leave this. My admiration for the 3 or 4 people. One of them is Amresh Kumar, and Vijay, whose work was also in Malika's presentation. See, I think he's among the highest published person of his age. He's publishing something like 12 to 13 papers a year for the last 10 years. Excellent high throughput. And that department of 3 or 4 faculty in Manesar, I have to admire Professor Rajendra Prasad. I mean, the 5 years between 2005, 2010, he changed the face of JNU as the Pro-Vice Chancellor because of the way that he channelized infrastructure towards the then young faculty, myself included. And our throughput went from about 3 or 4 papers a year to 10. And I would say that JNU in that decade really rose to number 2 in the university space, not because of its reputation, but because of its output, because of that kind of planning. And I really look forward to the way that this data science program will evolve. I have the highest expectation for it. And Ravi, I'm sorry I couldn't hear you, but I would look, the next time we talk, I will definitely make the time. I apologize for Professor Prasad, my lack of communication, but you can always call me and I will always pick up. Thank you, Dr. Selvamoorthy.
Thank you, Dr. Andrew. And
Professor Selvamoorthy, just one sentence.
Yeah, please. So fortunate to hear good things about you and also Amity. Thank you so much. Rajendra Prasad, please.
I feel sad that Ravi was all dressed up with a tie and everything. So he has to go back. But Andrew has covered a lot. And I must add to dignitaries who are listening, our data science program is one of the most popular program for data science MSc. And, we are overwhelmed with the response and we don't have space to admit more people. And these guys have established, and thanks to Andrew, he has been a brain behind. and most of the faculty which have come to MIT Manesar is his product. So it is his family, the clonal variation of Andrew's in Manesar. So we are very proud, and he's always available for our help as expert and otherwise also. Thank you, Andrew, very much. Thank you.
Is Dr. Ravi Datta there? Can we just see you on the screen?
He's there. Poor. Ravi, you can show your tie.
Here.
Ravi? Oh, Ravi. Let's clap for Ravi. So much appreciation from Dr. Andrew, Rajendra Prasad, and we are proud to have such youngsters driving MIT today. You are the future. You are the present and future of MIT and the India and globe. So you'll do wonders. We'll listen to you next week positively. You will be the first speaker. But we want to have you physically present here, not on online, because Gurugama and Amity is not far, Noida. So next time, please come. We'll fix up a convenient date after taking time from Dr. Andrew and Dr. Rajendra Prasad. Then
I promise to bring Andrew on the campus when you fix
this. We want to show him the campus, meet him physically in person. And Dr. Andrew, you're wonderful. You are helping so much. What Amity is today, not only these people, but people like you who have blessed, supported, guided, mentored, facilitated, catalyzed our growth. We need your continued support, and certainly we'll be looking towards you and JNU in a big way. We want to sign an MOU with JNU. Rajendra Prasad, I have discussed this with Vice Chancellor.
Yes.
A new lady Vice Chancellor.
We should move one day together or something. Yeah.
So we should now take it forward. Thank you so much. Now I would request Dr. B.C. Das to give concluding remarks. And I learned a lot. I enjoyed listening to these youngsters. This charges me. This makes me younger, energetic, enthusiastic. And so thank you for all the speakers from my side, that you are doing great job. And India is moving on the right path because of you people, youngsters, who will see the Amrit Kaal, who will see India growing as a developed nation, and who would see India assuming leadership position in science and technology in various domains. So that is why you will do that. All of you, the path-breaking researchers whom Amity has, network with people like you and other experts who joined us. Rajendra Prasad is bubbling to say something.
I know Mona Lisa work I was knowing a bit, and she's of course wonderful researcher. But this Durgesh and Malika is a dynamite. I mean, what they're doing is really, they will go a long way, and we're very proud that they're in our campus, really. And would like really to invite them to our campus just to see that what is going on here and interact with others. Malika, you are not listening.
Right. Yeah, thank you. Thank you, Dr. I want to listen from Dr. Neeraj Sharma and Dr. Chakravarti 1 minute. They have been listening to all the presentation. They are the SNT war room experts and who are driving.
Rajendra Prasad sir, thank you.
Oh, okay, thank you.
Mona Lisa this side.
Okay.
I was sitting at
I
the
will
back.
start with Chakravarti.
No, I think it was like, as if I was in an atmosphere and ambience where science is being discussed for its possible applications. I myself was not knowing that MIT's future is so bright with these youngsters making us proud on a daily basis. Malika winning the Fulbright Fellowship is also a testimony. We missed Sharma today from Gurgaon, but I am pretty sure his presentation would have been equally dynamite as that of Mona Lisa and Durgesh. So I am quite optimistic after today's session, sir, and the founders' desire that can we convert them into wealth will be a task, challenging task, which we would be taking up to the creation of startups and connecting, as per Dr. Selva Murthy, connecting Lakshmi with Saraswati. Thank you very much.
Dr. Neeraj Sharma. See, these are people who came from our Ministry of Science and Technology. One from DST, one from CSIR. They have assumed very great positions. So founder president has been able to attract such brilliant people who are building Amity today. Neeraj.
So I second what Dr. Chakravarti has mentioned. Our task is a little difficult now, as Honorable Founder President has entrusted us. I would only request that such presentations, which are beautiful presentations, and a lot of new things are happening here. We need to get more time, so we will have to discuss with them individually, personally, and talk to them how to convert their ideas into real startup companies. It's a long process, but we need to discuss and talk. But beautiful presentations that they made.
Thank you, Dr. Neeraj. Now over to Dr. Das, who is the Dean of our Health Sciences and also the Chairman of URC. Over to you.
Thank you, Dr. Selvamurthy. And after the long last, so I could not attend in the beginning for an hour or so, but then I was listening to it. I was in the clinic, my visit to the hospital, so it was due, so I could not there, but I have listened all the talks. First talk, the three speakers, but before I go to the speakers, but it is, I must thank and all gratitude to our Honorable Founder President, because this path-breaking research and contributions and in the Amity group of scientists, that is the brainchild of our Honorable Founder President, and that has been taken forward by Dr. Selvamurthy and the ST group of people. So that's, I think it is the third or fourth one. Fourth one is going on and very successfully, and finding out those who are the excellent researcher, and it is just a discovery in the youth populations. So in the Amity populations of the faculty. But first of all, I must thank all the three speakers. One could not be spoken today because of the lack of time. So we have decided, as Dr. Selvamurthy said, that we will have two talks so that we have sufficient time and not in a hurry, and we can discuss and have a, but also thanks to all the experts who have come physically, but or attend through online. So I thank all the experts, those who have contributed, given their expert comments and how to go ahead of their excellent research. So I must congratulate all the three speakers who have spoken their individual area of specialization. That is, first is the chemical sciences, and the most recent are the frontier areas of the click chemistry or green chemistry, and along with the nano science. And the second is the plant sciences, and he has shown an excellent contribution from the publications and all. And also he is the 1% of the world scientist. So with this AP3 level, so that's a great inspiration for others also. So I think all the speakers are here or not? You are there. So I was searching for you because I have several comments reserved, because I always take things while I praise endlessly to their efforts, but at the same time I'm critical of many of the things, the findings and the publications and all. So I will just briefly share. I will not take 1 hour to speak, so that will take a lot of time, but certainly, so please have your seats. So the Malika, not Malika, the Mona Lisa started with she mainly focused the graphene quantum dots and exhaustive nano structure for the development of many other things, including the metal organic framework, MOF, which is mostly, and it is the frontier areas, are used for drug delivery and many biomedical or biomedical engineering aspects. So one thing I would say, your presentation was excellent. I was listening, but the things which came out in the last talk also, that you have to bring it to that how it is applicable, deliverable, how it is, because all are not hardcore scientists. There may be some junior research scholars or something, and they are not maybe the same field of specialization, because as I told, it's a chemical sciences, then plant sciences, then neurosciences. So all are not the specialists on this field. So one has to bring it to some deliverable, that how it is to be utilized. So some point of the time, at the end or in between, you should bring in that this is the study which is will be applicable. Like in Mallika's case, she has been speaking very hardcore science, the neuroscience and molecular biology and all. But then it has to bring it to, when she was asked, then say, she is fantastic. And your science, those who are in your field, they will highly appreciate. But then those who are not of the field, you have to bring it to the delivery or all the biological functions. So that has a huge application in the disease system. So how it is, it can be extrapolated, or how can it is applied in the disease management. So these are few of the things, but then Dr. Monali says the comprehensive utilization of your this technology for the product development, and then the application of the biomedical science, particularly in the disease management. So this is very important, so that we know that you have been working, but this is to focus. So this is not to add here. Please don't add. So it is good. You are doing excellent work. Everybody knows, and it is translation. So translation science, we look forward something, not that immediately we expect, but you have to predict that you are working something which will come in a near future or distant future.
Yes, sir. Meenakshi is trying that stem cells part, that without.
You told. I have listened to you.
We are trying, sir.
I have listened to this talk.
Yes, and that I got inspired after discussing with you, working with your group. And
so keep in mind that which can go to the society or to the humanity. So that is one thing important. I think our medical friend from the experts from outside, so they have indicated that. So that is very important.
Yes.
So you are all doing excellent in your field of research. I come to Durgesh Tripathi. So I feel proud that you are all the BHUites. So you have a connection with the BHU. You took that. I am also from the BHU alma mater, my BHU. So good. But your university is very good. But I know this silicon. I am a biomedical scientist. So I have that's silicon is, which I think Nutan Kaushik told something, that you see, like in the plant or wheat, we use many things, zinc, and then oil value, and many things we add. But then these medical friends will accept that what extent you will add. The silicon is a problem, but it is also good. As you said, that it is a nanoform. If you use, it can be utilized in a fertilizer, good fertilizer for growth of the plant. But it is in a nanoform, it's a nano quantity. But then in the plants, in the rice and the wheat, it is its accumulation is heavy accumulation. And that is why that was my idea. That is how you can utilize the problem of the country, solving the country. You have indicated, but you have not come out with any indication of the solution. That is the stubble. How the stubble, nobody is ready to use but to burn it, because it has a heavy content of the silica. But that cannot be used by anything. So I appreciate your head, the director. He told, I have had the microbial enzyme which can degrade the silicon. So you can join hands. It could be an excellent discovery rather, if you can make it, which can go for a startup also, an industry, and use that so much of problem and pollution created by the stubble.
Yes.
But if you can downgrade you sit down. So this silicon, so the heavy silicon, but even in the plant system, you said that it is in the human, if it is used by these seeds and all, we use but then the rice or the wheat and all, but that creates the lining in the blood vessels.
Yes.
So that will later on lead to the contribution to those who are cardiologists will be knowing that they may create a problem. It may be you are telling it is for the better movement, but but it may cause a pathological condition also because they are toxic. So to certain extent they are not toxic, but certain above the toxic, anything is too much. It is a general knowledge.
Right.
So it is, if it is more accumulated, then it is toxic.
Yes.
So that is why stubble is not being used by anybody because it has a huge silicon. But how to reduce the silicon so that can be used? So these are few of the observations I wanted to share with you, and I think few more points.
Thank you so much.
The stubble I told. So so this is what the toxicity testing and toxicology, that is very important.
Right.
So anytime you can walk down, we can discuss also
Okay,
something.
thank you so much, sir.
Yeah.
Thank you
So thank you very much. So the Malika. So I know I have been looking after before that Malik joined this department. So I know her work. And first of all, congratulations for your Fulbright fellowships. So, but the presentation was excellent. And I'm happy that you are extending your PhD work, which is an excellent work, and done at a excellent institute in India and abroad. So that you are continuing to get into a pathological conclusion. So I think it is an important thing that what you start, you go deeper and deeper, so that you leave today and add something. And on the way of telling that, which it has been added by the experts, that it is all not that the HSPGS, the heparin sulfate, the protein glycans is not the all in all, that everything is that.
Right.
And also it is important to look into I will not take much time, it is taking time. So it is to look into if you can pinpoint some genes that related to the sleep deprivation and to the leading to the your SSPG or alterations. And you said something that I noted down somewhere. It is not seen here. I think, yeah, the RAB1. RABO1. No?
Robo1.
Robo1 and Robo2. So when it is Robo1 is downregulated by the transcription factor, that which is downregulated. Robo1 or Robo2?
Robo2 is downregulated.
Downregulated, but then
Robo1 increases.
Robo1 increases, but at the same time, they do the function of the Robo2.
It should.
It should. No, but you said that it works. It means, is there a homology with the Robo1 and Robo2 sequence homology?
Yes.
So that if that must be quite a few good homology. That's why they can take care of the Robo2 function.
That is known.
Yeah, okay, that is known. That is not due to mention. So I don't study, so that I have not studied that. That's why I had the idea that you can look at the sequence homology. So it is automatically your statement says that there should be homology.
Correct.
So this is, and also to this, that what I said, that you can look for some genes which are related to this activity that you are finding, whether you can pinpoint some genes, including your sleep deprivations. And it is known that when it's sleep deprivation, it has a connection with the brain only.
Yes.
We understand that in a simple biological knowledge. And it is, and there are huge number of studies. So the sleep and then light exposure and all these things, they are all related, and there must be gene involved.
Right.
And so that is important to find. That will give a good discovery rather.
Right.
And you can at the same time to find out some deterrent or inhibitors, some drug that what you are seeing, whether you can revert that.
Yes, sir.
So is there anything? And you have an excellent system of geographies.
Right.
So anyway, somebody asked that why you are using geographies. So it is an excellent system. So people say, why are you using Drosophila? So, but one important thing that how much you can extrapolate.
Right.
That has to be seen. And that extrapolation to human, I think my colleagues said that it is important to extrapolate to human. Yes, that is what I wrote also. So he said that. So extrapolation to the human system is the most important thing. Ultimate, you have to go to the human.
Right.
So this is all this research is fine, but then to understand, we are using the system to understand better way, because we cannot many things can do in the human system. That is tough and very difficult, and ethical clearance and many things are there.
Right.
Yeah, so that's why we take.
Like he pointed out, organ on chip is one very expensive at this point, but a very viable technique compared to like right now we are only limited to postmortem samples.
So
that's
I
a
think
very good.
I will not extend much. So I will. So these can be neurodevelopmental disorders. How can be interpreted in the human neurodevelopmental disorders, and can be used to find a gene or any marker or a protein, so that whether that can be used as a diagnostic marker, early diagnostic.
Early diagnostic marker.
So that can make a lot of difference.
That's a goal, yes.
And then you can find the deterrent to that or inhibitors to that. If you understand the pathway and the pathway analysis, if you find the gene, then you can enter into the pathway analysis and the bioinformatic search. Many things will help. And then you can have the validation. And then.
Yes, sir.
So these are few of the observations. So though that is, I thought I will add into the concluding remarks, but nevertheless, I congratulate all the astute people, including the first is the Goodwill and others.
Goodwill, stand up. Let's give a big clap to Goodwill Poker.
So she is a very silent coordinator and very strict coordinator. She does many things, not only this pathbreaking, but many other things, and along with the others in the division.
I think Vaibhav, you also get up, and all the debaters.
All are
Tiwari.
Yes,
Very good.
Tiwari is everywhere, omnipresent.
And above all, Gopal Bhushan. It's good. Gopal.
So, thank you. Thank you all. And I thank Dr. Selvamurthy. Nevertheless, he is the guiding force behind this, all this activity. Of course, the founder president is but last but not the least, all the experts, those who have most important that I endeavor to do in favor of the Amity University, all fraternity, that they could come and join us and physically. And also those who have on online, I thank Professor Andrew as well as Professor Rajendra Prasad from JNU as well as in our Amity campus, Gurgaon. So thank you so much. And I'm sorry we have that this the last candidate could not present this time, but nevertheless, we will listen next time. Yeah.
Presentation.
So thank you very much.
That's the good presentation. No,